Wonderful. Thank you all for joining us today. Good afternoon. We have people online from all over the world, so good morning and good evening and good night. Welcome to the 2026 Science Meeting and Research Update from Friends of FSH Research. I am Amanda Rickard. I'll be your host today, and I'm also a volunteer and member of the Friends Board of Directors and Scientific Advisory Board. And then I also work as Managing Director of the Global FSHD Innovation Hub, a subsidiary of the FSHD Society. So pretty much eat, drink, sleep, live, working toward a cure for FSHD. I'm fortunate to have benefited from one of the areas of focus for Friends overall, which is this idea that we fund research projects whose purpose is to get new organizations, new investigators, new students interested in this disease long enough to get our hooks in, and then hopefully inspire and support their long-term dedication to this community. And we'll hear more examples of that today, and also in this program, and also tonight at the auction as well. So this year's update is focused on genetic medicines and gene therapies for FSHD, an area of a lot of progress in recent years. Just so that you know, our format will include a video recording of each presentation, followed by a live Q&A with each speaker. Some of our speakers are here in the room and some are on Zoom. If attendees here in the room have questions for the speaker, the easiest way is if you could line up and I'll hand the mic to you and you can ask your questions. If that's not possible, I can bring the mic to you, so just raise your hand. And attendees on Zoom, please enter your questions into the chat. We'll do our best to get through as many of those questions as we can. Please bear with us. We're running a pretty tight ship. Terry says we're not allowed to be late to the auction, so we're going to move as quickly as we can. And just so that you know, this program will be recorded and shared via YouTube and through the Friends newsletter, so you can go back and watch it there. So before we get started with the videos, I'd like to first thank our sponsors of today's event. That includes Sanofi and the Muscular Dystrophy Association. I'll also introduce my colleague and friend, Dr. Jeff Chamberlain. You may have recently heard a sigh of relief from the Seattle gene therapy community as we heard rumor that Dr. Chamberlain was tentatively had accepted a position as director of gene therapy at USC Med School, but ultimately, as far as still true, ultimately decided to remain at UW for another 5 years. So good news, we're safe for now. Yeah. Worth celebrating for sure. Dr. Chamberlain is the fearless leader and chair of the Friends of FSH Research Scientific Advisory Board. He's a professor and McGaw Chair in Muscular Dystrophy at the University of Washington's Institute for Stem Cell and Regenerative Medicine. And he is director of the Seattle Wellstone Muscular Dystrophy Specialized Research Center and the former president of the American Society for Gene and Cell Therapy. So please join me in welcoming Dr. Chamberlain.
Thank you, Amanda. Well, it's a real pleasure to be here today, and I got to say though, you know, when I got up this morning, it was 24 degrees at my house, and I started thinking, you know, Los Angeles maybe wouldn't have been so bad after all. But at least I'm here. I'm happy to stay here, and I'll be able to still root for the Seahawks tomorrow against that horrible Los Angeles team. So we'll see what happens with that. But it's a real pleasure to be able to be here and be part of the research forum today, and to be able to introduce some of the what I think are going to be fantastic talks about work that's been done under the support of the Friends of FSH Research. And you know, the research that we're going to hear about today, again, has been supported by Friends. It's who have done just an amazing job of really bringing this disease into the forefront and jump-starting a lot of work. You know, my own experience dates back a little over 20 years now, I think it is, since Terry Colella decided to get heavily involved in supporting research and clinical care with FSHD. And what happened, you know, about 20 years ago, she was having trouble finding people to actually do the research and to get involved. And she came over to the University of Washington and met with a couple of us, particularly with Stephen Tapscott and with Tom Bird at the time. and convince them to start getting involved in research. And that was about the same time as we formed the Wellstone Muscular Dystrophy Research Center at the University of Washington. And at the time, I'm almost embarrassed to say, the Wellstone Center was focused entirely on Duchenne muscular dystrophy. But through the hard work and dedication and persuasive skills of Terry and Rick Colella, the research focus of our center has been shifting over. And now I think it's probably about 60% focused on FSHD and 40% focused on other muscular dystrophies. So that's been a transformational event at our institution, and again, it's really entirely because of the Friends' support and encouragement. Over the last 20 years, not only this research symposium here, but also particularly the fundraising event that hopefully everyone will be at tonight, has raised millions of dollars for FSHD research. Initially, some of that money was coming in to the University of Washington to jumpstart efforts there, but it's expanded enormously, and as we'll see that today. We now have researchers throughout the United States working on FSHD. Many of them that have been supported by the Friends of FSHD, and we even have groups throughout the world now, in Asia and in Europe, that have been not only working on the disease, but working closely with Friends and submitting grant applications. And George Shaw reminded me earlier, we're about to receive a new set of grant applications at the end of this month that we'll be diving into, and I think we have a record number of applications that have come in. And so that's good news and bad news, because the more people that are involved and interested, that's good news. The bad news, it's going to be hard to sort out the cream of the crop and decide who to fund. And so hopefully we'll have a great event this evening and bring in plenty of money so that we can support as many of these grant applications as possible, because there's nothing more fun than being able to tell people that they've received a grant, that we're going to award a grant to them, and also nothing more sad than having to tell the other people that they didn't quite make the cut this year. So hopefully we can minimize how many people aren't funded. Just briefly, we'll hear about a variety of different research projects. I can say briefly, as Amanda alluded to earlier, my own laboratory has been focused on gene therapy for many years. It's still a little unclear how well gene therapy might impact FSHD, but it is having an enormous impact on many other muscular dystrophies. When we first started working on gene therapy, most people I would talk to would just say, "Why are you doing this? Gene therapy will never work." It is starting to work. We still have a lot of work to go to get it working better, but there are now 13 approved gene therapies in the United States alone that are out there and being available by prescription drugs. And there's a little bit of work trying to adapt that to FSHD by a number of the grant applications we get. I think some of the talks we're going to hear today are focused on gene therapy approaches, and Joelle Chamberlain at the University of Washington is focused on a gene therapy approach for FSHD. So we'll see that come along. But there's also many other approaches, looking at small molecules, antibody-directed therapeutics, and still a lot of work going on in trying to really understand the progression and the time course of symptoms and what is really going on in the slowly progressive disease that we need to reverse. So without further ado, I'll maybe turn it back to Amanda, and we can start hearing about the research. So.
Thank you, Jeff. Okay, our first presentation today is from Dr. Gregory Block. Dr. Block is co-founder and chief executive officer of Histone Therapeutics and a member of the Friends of FSH Research Scientific Advisory Board. He has held executive and leadership positions at Universal Cells, Astellas Pharma, and Notch Therapeutics, and now leads Histone Therapeutics, where his team uses precision epigenetic reprogramming to enhance cell function and treat disease.
Hey everyone, I guess if you're watching this, you're probably at the Diamonds and Denim Gala Research Update 2026. This is amazing. We've been funding research at Friends of FSH Research for quite some time, and I'm feeling the impact of this year. I'm giving you a research update on what is, I guess, over 15 years of being supported by Friends of FSH Research. I received my first grant from them back in 2011 when I was a postdoc at the University of Washington. And fast forward 15 years, we're now running a company focused on FSHD, using everything we've learned from studying FSHD, and we couldn't have done any of it without friends at FSH Research. The team has asked me to give a bit of a background on genetic medicines. We're going to be hearing a lot about them. They're kind of hard to wrap your mind around, and so I thought I would spend some time just distinguishing the difference between what a genetic medicine is and what a gene therapy is. Genetic medicine is quite simple. It's just a medicine. Instead of it being a small molecule or a biologic like an antibody, it's a medicine based on RNA or DNA. So it's literally a sequence-based mechanism for the drug. They can encode proteins, they can block proteins. There's all sorts of nifty things that you can do with these types of medicines. But the key distinguishing factor is that they're made of the same genetic material that is used in our cells to encode proteins and genes. Now, gene therapy is a type of genetic medicine, but there are differences. A gene therapy is usually permanent. If you have a mutation in a gene, for example, perhaps you can replace that mutation in the gene with a working copy of the gene. This usually works by altering the sequence of the DNA or perhaps altering the chemistry around the DNA. But it's usually, you think about it as like a one and done therapy for curing a disease or reducing the burden of a disease. Whereas a genetic medicine works a little bit differently. There are a number of approved genetic medicines on the market. They tend to be redosable. They work just like a medicine. You dose them, you figure out the frequency of dosing. It can be stopped if the therapy is not working. And they are, while they are still based on the fundamental mechanism of being a nucleic acid or like a DNA or RNA molecule, they tend to be a little less permanent. And sometimes the lines can get blurred. So it can be a little bit confusing when you're reading exciting things happening in the industry or in the field to distinguish between what is a genetic medicine versus what is a gene therapy. We had a major amazing thing happen in 2025 in our industry. Baby KJ, a child with a severe form of a disease called CPS1 deficiency, received a personalized CRISPR medicine tailored to correct their genetic mutation. This used a very short-term expression of a CRISPR molecule. So it was a nucleic acid medicine delivered to the liver, but that then introduced a permanent cure into those cells. So they used a genetic medicine to deliver a gene therapy, and now baby KJ stands a much better chance than they did before. It's a really exciting thing for our field. And with this context of genetic medicines versus gene therapy, why don't I tell you a little bit about what we've been building at Histone Therapeutics, a company I co-founded with Shiri Levy at the University of Washington, which focuses on the biotech worldview of everything upside down. Instead of trying to block things, instead of trying to stop a genetic mechanism, we think differently. We think about gene activation. How can we turn on genes in order to treat disease? And why would you care about turning genes on? Well, if you think about it, every single cell in your body has the same DNA, and that DNA runs fundamentally different programs. Those programs are usually attributed to only about 10% of the available genes in your genome. What about the other 90%? What if you could reprogram a cell to do the thing it should be doing but otherwise isn't, based on the repertoire of genes that already exists in your cells? If you did that, if you had the ability to access these genes which are otherwise silenced, you could essentially reprogram cells into a state of existence that otherwise didn't occur. Now, why don't people already do this? Well, we've been activating genes for decades in the lab, but they tend to be useful only for activating genes which are already poised to be active. So, for example, this on this slide, I'm showing a schematic of a thing called a Viper protein, or VP64. This activates genes which are essentially already active. And right now, there are no genetic medicine compatible versions of a gene activator that can activate silent genes. We've invented one. We call it 3R-on, and 3R-on is particularly good at activating genes which are already silenced. And to design 3R-on, we really had to go back to some fundamental principles, because if we're going to create this platform that can do this wonderful thing, we wanted it to be safe. We wanted to essentially be reversible. We wanted to be potent, so we don't need insanely high doses. And we wanted to avoid enzymatic activity, which is a bit of a nuance that I'm not going to go into details. You can ask during question period. But essentially, we sought to create the world's safest genetic activator that can activate or drive transcription through silenced genes. And this little graph I'm showing in the bottom here is actually the ratio of fold change between 3R-on versus the benchmark Viper protein on a silenced gene. It knocks it out the water. It works really well. We oftentimes detect activation of genes below the threshold of detection of the things we delivered. So it's quite a potent genetic activator, which we believe we can apply to genetic medicines. And if you think about genetic medicines as a redosable piece of DNA or RNA that can be used therapeutically, like I described a couple of slides ago, this puts us squarely in the category creation of redosable upregulation, which is a very interesting way to think about the world. There's a number of different diseases that can be treated by genetic upregulation. And if there was a modality that allowed us to do this in a redosable format, this could be a really successful company. We know this technology works in the context of muscle cells. It's quite interesting. We've shown that we can activate therapeutic genes in muscle, and we've shown that the specificity is unprecedented. So, for example, in this slide, I'm showing the activation of a therapeutic muscle gene in muscle cells. These are mouse muscle cells. In the purple bars, you can see the gene expression is screaming when we put in our 3R-on molecule. But when we do the same thing in the liver cell, it does absolutely nothing. Now you'd think to yourself, is that a good thing or a bad thing? Well, it's a good thing, because what it means is that we're able to create an activator that is not promiscuous, meaning it's not going to work everywhere. We have to dial it in, and that will help us formulate an actual promising regulatory package when we go to move this thing into patients. We wanted to test this in vivo, but we realized we didn't have the equipment to do so. So enter Friends of FSH Research to support us again. With the Flex Fund that's available from Friends, we were able to purchase a syringe pump system that's in our labs in Seattle. This is for formulating genetic medicines within lipid nanoparticles. This is available to everyone in the research community to use, and we hope that you reach out and let us help you make lipid nanoparticles. It's really fun to do. Anyways, we made lipid nanoparticles containing mRNAs, encoding 3R-on, targeting the gene that we wanted to be therapeutic in skeletal muscle. This was just a platform validation experiment that we did. It's not relevant to the work that's been funded by Friends so far. But with FSHD Canada, we were able to test this in vivo to see if it even works. Does this thing that we've invented even work if you inject it into a muscle? And the answer is it works really well. We see really nice fold change of the gene that we're targeting in the muscle. And kind of like what I showed you in the previous slides, what happens in the liver is negligible. So the liver is important in this context because that's where the lipid nanoparticles drain. And so what that means is we could essentially create a therapy that's specific for the muscle but doesn't cause weird things to happen in the liver. So we need to think about FSHD now. The goal in FSHD, as you'll hear a lot in these presentations, is to either enhance muscle strength or to enhance muscle generation, but most importantly, to deal with the disease-causing gene DUX4. Now, if you were a disease silencing company, you would say, well, I'm just going to go silence a DUX4, but we're a gene activation company. So what we're doing is we're looking for genes that we can activate that can reduce DUX4 expression levels. And what's interesting is we know from patients that there are other genes in the genome that if you have differing amounts of those genes, you have a different phenotype of FSHD. Some phenotypes of FSHD are more severe, some are not. If we can upregulate some of those genes that are mediating that, we could potentially make a drug here. And so in our collaboration, which was funded by Friends of FSH Research, we worked with Fran Sverdrup at the University of St. Louis, or St. Louis University. And we screened a number of different genes. This was not an easy project, but we did find a gene that if we upregulate it, shuts down the DUX4 program while retaining the ability for skeletal muscle to still differentiate. For the researchers in the room, this is obviously one of the nuances of running these type of assays. You need to make sure you're not killing the ability for the myotubes to form. And in this case, the gene that we're using seems to enhance it. So we're liking the way this gene is looking, and we're validating a lot of these studies in the labs at Histone now in Seattle. We received a second grant from Friends of FSH Research directly to Histone this time to actually work on the lead platform to understand it better so that we can drive this towards a therapeutic candidate. And one of those things was understanding the 3-RON molecule itself. So we put this into cells, we see potent gene upregulation. One could surmise that maybe if you have something so potent and you're putting it in a cell, it's a bad thing. But it turns out when we put 3-RON into a cell to activate a specific gene, that reaction is enormously specific. This data made us really happy. This forms the basis for understanding how we move this forward into animal models and into the clinic. And if you want to create a redosable gene upregulation-based medicine, this is the exact phenotype of a gene expression profile you'll want to see after you perturb the system. So where do we go from here? Well, we want to understand 3-RON more. We're actually working with a machine learning specialist who's helping us see if we can predict where it will activate genes and where it will not. And then we're going to take a couple of things into animal models. One, we're testing our in vivo DUX4 reduction strategy, which I just showed you in the previous slide. But we've also been working at the company on metabolic health. And the more we understand that, the more we think to ourselves, this could actually be a standalone therapy on its own, where we've shown that we can upregulate master regulators of global systemic metabolism, which may have a massive impact on patients with FSHD. And we're excited about where that research can go. This is the team. You'll see many of us at the auction tonight dressed in our denim jackets and hopefully cowboy hats. We're a small company still, but we are growing. Shiri and I co-founded the company, and we run the company, me, Shiri, and Alan. We have some very talented muscle biologists working in the lab. Jesse Macadangdang, Nate Cunningham, and Georgia Mewis are helping drive this program forward. And we have wonderful scientific advisors, some of which are very familiar to our community. Bruce Wentworth, obviously a founder of Dyne Therapeutics, has worked on antisense oligos throughout his career. And we are building a lean, mean machine for developing new drugs for FSHD. It was wonderful to talk to myself alone in a room. I hope this is not super embarrassing to watch once I'm in the room watching it with you. And I guess the real life version of me is going to take questions from you right now. Thanks very much. Bye.
All right, let's give Greg a round of applause. We have two speakers who get to watch themselves recorded and then answer questions live, so that's probably the bravest thing anyone's ever done. Yeah, Greg, you want to come up and we can take questions up here? Yeah, well done with the continuity on the jacket, very nice. One of my questions for you was, did you wear denim in your video on purpose? So clearly the answer is yes. Okay, do we have any questions from folks in the room? Yeah, I'll run this mic over to you, 1 second.
I have a very basic question. The tissue targeting is happening because of the lipid particle is the targeting agent, or does that happen at the level of the 3-on activation?
Yeah, that's a great question. In this case, we're using lipid nanoparticles that are not targeted. They're not modified in any way. If you were to inject these lipid nanoparticles IV, they would basically drain straight to your liver. These were proof of concept experiments, and we just needed to get them done. And we're learning how to do lipid nanoparticles right now. So the muscle specificity came from injecting it into the muscle. And so we were able to see the activation in the muscle, but we also could see the 3-RON molecule itself. We could detect it in the liver afterwards, but when we looked in the liver, we didn't see the activation of the target gene, which was kind of interesting. So in a way, for this particular gene, we showed we can get tissue specificity governed by the targeting mechanism, governed by the mRNA, which was very unique.
I think it might be worthwhile to explain what a lipid nanoparticle is to this audience.
Yeah, so lipid nanoparticles became famous during the pandemic. It's the basis of the Pfizer and Moderna vaccines. So they're little balls of fat that when they nucleate around an RNA, and it allows for a cell to take up the RNA, and then it goes through this. So I picture the Lord of the Rings where they have to go through all these horrible traumatic experiences just to get out. And then that's what happens to an mRNA. It has to go through the cell and survive. And then if it gets through all that, it makes a protein. And that protein can create a vaccine. Or in this case, a thing that goes into your nucleus to activate a gene.
Okay, we'll take one more question from Tessa.
That was great, Greg. My question is, so DUX4 targets are fairly long-lived in the cell. You showed mRNA expression of some DUX4 targets. Have you guys looked at proteins? Because the proteins can last for a while. So are you thinking of doing maybe a time course after your 3-RON to see that you get also downregulation at the protein level?
Yes, we need to do that. We haven't done that yet. We had a hell of a time actually getting stuff into the muscle cells. So that took a bit of time, and so these are next steps. We've got to get the system up and running with the lipid nanoparticles in the labs, and start to just look at this from all the different angles. This is basically the first encouraging blip that we saw.
Okay, great. Let's give Greg a round of applause. Thanks, Greg. Okay, next up we have a presentation from Dr. Scott Harper. Dr. Harper is a PI at the Nationwide Children's Hospital Center for Gene Therapy and a professor at The Ohio State University. I feel like I'm introducing NFL players. His lab studies the development of RNAi-based treatments for dominantly inherited neuromuscular disorders and has received multiple grants from Friends of FSH Research to support their important and innovative work. He is also a co-founder and chief scientific advisor of Armatus Bio, a biotechnology company leveraging the power of vectorized RNAi to precisely target the genetic drivers of neurological and neuromuscular diseases.
Hello, my name is Scott Harper. I am a principal investigator at the Center for Gene Therapy at Nationwide. Let me start over. Hello, I'm Scott Harper. I'm a principal investigator at the Jerry R. Mendell Center for Gene Therapy at Nationwide Children's Hospital in Columbus, Ohio. I'm also a professor of pediatrics at The Ohio State University, also in Columbus, Ohio. And a few years ago, I co-founded a biotech company called Armatus Bio that the goal of which is to translate some of our gene therapies for neuromuscular disease, including FSHD. And today I'll be telling you about the topic of my talk, which is gene therapy for FSHD. And here are my financial disclosures. I am co-founder and chief scientific advisor at Armatus Bio. I have patents, some of which have been licensed by the company, particularly on FSHD. And then I have some other scientific advisory board memberships for the CMT Association and Externa Bio. So today I'm going to be telling you about gene therapy. And so what is gene therapy? I think it's good to start with a basic definition. And what we mean by gene therapy is treating disease with nucleic acids. And what nucleic acids are DNA or RNA. And in particular, we're using both types of nucleic acids to develop this therapy. Gene therapy is it's been around for a while. The concept has been around for a while. Here's a paper from Science published in 1972, where the title of which is Gene Therapy for Human Genetic Disease with a question mark. And so at this time, the gentleman who wrote this paper were postulating that gene therapy might be possible. And today it is. It's no longer science fiction. Back then in 1972, it was just a hypothesis that we could treat disease with DNA or RNA. And now we have a number of different gene therapies FDA approved in the United States and also equivalent agencies in other parts of the world. So when we're talking about gene therapy, what we're actually talking about here is what ideally would be a one-time treatment. And so the idea is that if we can deliver our therapeutic nucleic acids, DNA or RNA, to, in the case of FSHD, muscle cells, and deliver a therapeutic payload to those muscle cells that can correct the disease, if we do this in the right way, theoretically, we could provide lifelong protection. Now, this sort of experiment is ongoing now in humans, where a number of people have been injected with gene therapies similar to what I'll be describing here. And we don't know how long they'll last, but current data suggests that it could be decades. So I say lifelong, but that's theoretical at this point. We'll know a lot more about how long gene therapies can last in muscle in the coming years. So gene therapy is based upon the central dogma of molecular biology, and that's displayed on this slide. This is if you've taken high school biology, you probably are familiar with this. What this describes is the flow of genetic information in our DNA, in our genes. And so humans have about 3.2 billion base pairs of DNA. That's what we call the human genome. And those are encoded in 23 pairs of chromosomes. And on those chromosomes are about 20,000 what we call protein coding genes. Now, proteins are the things that make up our bodies, our hair, and, basically everything is made out of protein. And so we have about 20,000 of those in our DNA. And so what happens is in the central dogma, we have these genes encoded in our DNA. Those genes are copied into what's called a messenger RNA or mRNA. And the mRNA then serves as a template to make protein. And that's just indicated here. So when we talk about the central dogma of molecular biology, what we mean is this flow of information, DNA makes RNA, makes protein. Now for FSHD, what this means is, how does FSHD fit into this? Well, FSHD is linked to this gene called DUX4, DUX4. So DUX4, we all have it in our genes. And so our DNA encodes this DUX4 gene. DUX4, the gene is copied into a messenger RNA, the DUX4 messenger RNA, that then is, again, used as a template to make protein. And if that protein is produced in muscle abnormally, it causes FSHD. So our FSHD therapy and others in the field is designed to silence the DUX4 gene. And we're doing this at the level of the messenger RNA. And that's indicated here where we are trying to attack the DUX4 messenger RNA with gene therapy. And by doing so, we would destroy that messenger RNA. So at this step of the central dogma process, and then we would block the messenger RNA from being made into protein and therefore offer a treatment for FSHD. And so that's just summarized here. Our approach then is to develop a treatment for FSHD centered on DUX4 gene silencing that combines a process called RNA interference, which is what I just told you about. I'll tell you in a little bit more detail, attacking DUX4 at the messenger RNA level, and also muscle gene therapy. So the muscle gene therapy part is the delivery part. That's how we're getting our therapeutic to muscle. And I'll tell you in the next couple slides what both of these components are. So first off, let's talk about RNA interference and what that process is and how it's stimulated. So I mentioned at the beginning that we have these 20,000 or so protein coding genes that make up our bodies. But the human genome, that actually accounts for about 1 or 2% of the whole human genome. And so we also have, human beings, other genes called microRNA genes. And we have about 1,800 or so of those actually in our DNA. And a microRNA gene is different from a protein coding gene in that a microRNA does not make protein. Instead, a microRNA makes well, this is actually a structure. This is a rendering of a microRNA. It makes this hairpin shaped RNA structure, it's shown here on the upper right. And this hairpin shaped structure does not make protein, but what it does instead, it gets processed in the cell by a number of different enzymes to release what's called a mature microRNA. And that mature microRNA, so it's made in our DNA, and it exits the nucleus of the cell, and it can attack the messenger RNA at this level, and again cause silencing of that messenger RNA, thereby blocking the protein. So for FSHD, we can design microRNAs that can target this DUX4 gene and cause it to be degraded so it can never make toxic protein. And so this is sort of like finding a needle in a haystack, where the needle here would be DUX4 and our magnet here would be our microRNA, along with some gene silencing proteins. So, okay, we have this, and I'll show you data on this. We have this microRNA system that can derive or trigger a process called RNA interference against the DUX4 messenger RNA and cause it to be degraded. But we have to still, so that's one important component. Another important component of this therapy, though, is actually getting these nucleic acids into the muscle cells. That's actually really challenging. So there are different ways that we're doing it, and other labs are doing this, or companies even. So one way is you could deliver the DNA either as naked molecules, which actually that doesn't work very well, or you could conjugate it. And this is what a company like, for example, Avidity or Dyne are trying to do. They are using antibodies to couple to these DNA molecules, or actually RNA molecules in their case, and deliver those to the cell using this conjugated, what's called siRNA system. And I'll tell you a little bit more about in a second. We're not doing that. And in contrast, what we're doing is we're actually we're making a virus, or what's called a recombinant virus. So this is a natural virus that we can modify. We essentially put our therapy inside the virus, and that's shown here as this double helix. That virus has natural properties to infect muscle. And so it can get into these muscle cells and deliver this DNA payload that expresses our therapy that targets DUX4 and reduces its expression inside the cell. And so the viruses we're using are called adeno-associated viruses, or viral vectors, actually, or AAV. So you'll hear me say AAV quite a bit. And these can actually deliver a payload to the nucleus of a muscle cell. And once that gets uncoated, the DNA actually then becomes a factory to produce the therapeutic RNA inside the cell. And as I mentioned before, gene therapy is intended to be able to deliver a stable therapeutic product for essentially, theoretically, the lifetime of an organism, including a human being. At least that's the hope. So this is our approach. We're using an adeno-associated virus. We're packaging a therapeutic DNA inside the virus that then makes a microRNA that can attack the DUX4 messenger RNA and then hopefully cause a therapy for FSHD. So let me just show you the difference. I mentioned the difference between what we're doing with the gene therapy vector, but you can also deliver these RNAs using a non-viral system. And this is what, again, Avidity and Dyne are doing. Those are called siRNAs. And what I just want to point out here is that, okay, so let's start at the top. The microRNA here has a number of different components to it, but the arrowheads indicate processing sites inside the cell. So when we make this microRNA, it produces this hairpin, and then this hairpin gets grabbed by some cellular enzymes that make cuts, and they cut here and here and here and here. And essentially what that does, if you look at where it cuts, it releases what is essentially then an siRNA. So this is a natural process that all cells undergo. Every one of the 1,800 microRNAs that we have in our bodies is processed this way. Like, for example, Avidity's siRNA, they've essentially eliminated some of these intermediate steps that are required, and they're just delivering the final product as sort of a drug. So the idea is that both of these molecules essentially operate in the same way. They can both enter this RNA interference pathway, and they can cause DUX4 gene silencing. And we've compared the siRNA approach versus our gene therapy approach. And this is one of the benefits of what we're trying to do. I mentioned again that the muscle cells become a factory for producing the therapeutic inside the cell itself. So you'll have to deliver it once, and it will just continually be produced. Whereas the siRNA approach, and this is shown here, I'll walk you through this slide in a second, the siRNA approach is transitory. So it gets used up in the cell and then it goes away, meaning that after a few weeks you have to redeliver it. And that's shown here. We have our AAV, our microRNA we call MI405. So the blue lines are, we're just detecting the presence of the microRNA or the siRNA inside the cells after we've delivered them to mice. And what you can see is that when we have the same exact sequence, the same exact product, but instead we've conjugated it to the delivery system that, say, for example, Avidity is using. What you see here is that the siRNA approach starts out higher than the gene therapy approach, but over time it starts to go away, and by 6 weeks a redose is necessary. But our product continues to advance up to 6 weeks, and we have stable expression by about 6 weeks at high levels inside the muscle cells. So this is essentially the comparison of the two approaches, which is here's the siRNA. It gets into the cell. Most of the siRNA gets stuck inside a cellular compartment called the endosome. About 1 or 2% of it gets released, and then that can attack the DUX4 messenger RNA. And again, that has to be redosed. With our AAV system, the AAV vector can also get into the muscle cells. Most of that, again, gets stuck in the endosome and gets degraded. But the portion that gets released, the 1 or 10% with the viruses, goes to the nucleus of the cell, and it becomes a factory. And it will produce microRNAs continually theoretically for the lifetime of an organism, but essentially as long as that DNA remains in the nucleus. That gets processed and it accomplishes the same thing. So the siRNA approach and the microRNA approach using gene therapy accomplish the same thing by silencing DUX4 and therefore offering a therapy for FSHD. So let me tell you a little bit about the details of what we've done. Our therapeutic is called MI405, and this is an engineered artificial microRNA that we've designed to knock down or silence the DUX4 gene. I also want to point out that most of this work was done by Lindsay Wallace in my lab. The therapeutic mechanism, as I said, is DUX4 messenger RNA silencing. We're delivering the therapeutic DNA product that creates a microRNA using an adeno-associated viral vector, AAV. We're delivering this intravenously, and we're targeting skeletal muscle. And I just have a couple of slides of data I wanted to show you. We didn't just make one sequence, we tested a number of them. We tested 42 different microRNAs. First, we just did this in human cells, which we call in vitro screening. And so what this shows here is our microRNA MI405. The red sequence underlined here is the therapeutic component of this. This actually is capable of binding the DUX4 messenger RNA and targeting it for degradation. And so this just shows when we do the screening process that if we express inside human cells, our microRNA MI405, and we look for DUX4 protein, which is shown with these black lines on this, what's called the Western blot gel, we see that this is a control. MI-LAC-C is a negative control, so this shouldn't silence DUX4, and it doesn't. And then we have 3 microRNAs we've made to target DUX4, and you can see that MI405, our lead, essentially gets rid of the protein. There's maybe a little wisp of it here in this experiment. We've also tested this in muscle cells from FSHD patients, and so these are cell lines that if you let them grow on a plate together, they will fuse and they will start expressing DUX4. And we can detect DUX4 as these sort of brown dots here that you see in this muscle cell. So when we deliver our MI405 gene therapy to the FSHD patient muscle cells, we never find any brown dots. So we're concluding that we can eliminate DUX4 in human muscle cells. Now, we wanted to test this in an animal model. And when we started this project, there weren't any animal models that were useful in the field. Now there are several, including one that we made called tick DUX4. And I'll just point out two things on this that highlight what we think is one of the benefits of gene therapy. So first, I want to mention that the mouse model we've made really represents two mouse models. There's one that we call an uninduced leaky system that really just, we think that this actually models probably what's going on in human disease physiologically, where we have low levels of DUX4 expression and it takes a long time to develop disease in some cases. And so let's just start here. What I'm showing here, this is a muscle from the leg of a mouse that has been injected with a virus that's carrying our therapeutic, our AAV virus carrying MI405. But we've also put in a gene here called GFP that will tell you that the gene is present, or that the gene therapy is present by glowing green. That's what we're seeing here. And in this experiment, what we did was we took this low-level DUX4 mouse, this mouse model that's expressing low levels of DUX4, and we inject one leg with our therapeutic product, and we left the other leg untreated. And then these are muscle sections from those mice after a year. And so what we did was we took these at high power and then combined them so you can see a low power image of what those muscles look like under the microscope. Now you can't see any of the details here, but we intentionally colored damaged muscle blue in this image here. So what you see is that after a year in this mouse model, if in the untreated leg, we see all this blue area that's been damaged by DUX4 being expressed. And in the exact same mouse injected with our therapeutic, we are able to protect the muscle. We took this experiment out to a year. So at the end point of this experiment in 1 year, after one injection, we had complete protection from DUX4 damage. Then we also have another component of this model where we can turn on DUX4 at very high levels. We call this an aggressive model. And so within a few weeks, we can see muscle damage at low power and high power. And so this is a high power image here. The same kind of experiment where we had treated and untreated muscle in the same mouse. And what you see here is this is what normal muscle looks like. It's sort of this polygon shape, and the nuclei of the muscle, of a healthy muscle, are present at the periphery of the muscle cells. So each one of these is a muscle cell, and the blue dots are the nuclei. So this is what a normal muscle looks like, a healthy muscle. Damaged muscle, you see different shapes of the polygons, you see some presence of inflammatory cells, and you also see the nuclei of the muscle cell in the center. So central nuclei is an indication that a muscle has been damaged and is trying to repair itself. So when you see central nuclei, it means that a muscle has been damaged or injured. And so what we see in this aggressive model where we're expressing DUX4 at very high levels, treated side looks completely normal, and then the untreated side is damaged. And we think this is important because this aggressive model is probably unlikely that we see levels of DUX4 expression like this in humans. So the fact that we can treat these high levels of DUX4 in a mouse model suggests that this should be highly beneficial in humans as well. And so what is the AAV system that we're using for delivery was originally developed by Solid Biosciences. And they allowed us to use this new AAV vector they've made that is a muscle-targeted, it's called a muscle-targeted capsid. So this can express our therapy in muscle at relatively low doses compared to other gene therapies that are out there on the market now. So Solid granted us a license to use this for our gene therapy. And what we saw is, and I think I'm going to try to move quickly or more quickly through this slide because we're running out of time here. But I just want to point out that a typical dose for AAV, a first generation AAV system, is 1 e to the 14th. So that number doesn't really mean anything other than it's really big. And that's the dose that's required to hit most of the muscle. These are the doses that have been used in clinical trials for most gene therapy products today that use AAV vectors. And what you see here is that the SLB101 solid AAV capsid, we can dose at about 10 times lower and still get good gene expression of our therapeutic product. And we also see liver detargeting as well. And liver, what we know is that if the virus doesn't go into muscle, it goes elsewhere. And most of that, it goes into the liver. So we also see liver detargeting, which is a good thing because most toxicities associated with AAV deliveries occur in the liver. So we're increasing muscle expression and we're decreasing liver expression, which we think is going to be a safer approach. And just using this is the last piece of data I have, just showing that solids capsid outperforms the original systems we were using based on AAV, AAV9. And let's just compare here, AAV9 at high dose. This is the actual expression of the microRNA, our therapeutic product, inside a mouse muscle. And we have this little wisp of the microRNA that we're producing at this very high dose of our first generation vector system. But using solids capsid at 10 times less or more, we're seeing robust expression of our therapeutic. And we see, as a result of that, we see reduction of biomarkers, molecular biomarkers of FSHD in mice. And one of the ones we use is WFDC3. So we're able to suppress this biomarker that's associated with DUX4. And we also see protection of the muscle histology, so the way the muscle looks and functions. This is called an open field test. This is essentially a way that you can measure how far or how fast a mouse can walk. I'm happy to describe that in the question period. But what we see is that the saline treated, so in the red, so these are FSHD mice that are left untreated. They start to decline in this experiment, whereas the SLB101 treated mice perform just as well as what's called black 6, which is normal mice. These are non-FSHD mice. So our treatment then restores actual muscle function to normal levels in mice at relatively low doses in the E to the 13th range that have not been associated with serious adverse events in humans. And I'll just mention final slide here before I get to my conclusions. We've done this is a robust data set. We've dosed over 860 mice to demonstrate that this works. So to summarize, we've developed an engineered microRNA that we call MI405 that is able to trigger RNA interference against the DUX4 messenger RNA, causing it to be silenced and then causing a subsequent knockdown of the DUX4 protein. We're using a capsid developed by Solid Biosciences that is very efficient for AAV delivery to muscle. And we think it's potentially safer than some of the first generation systems that are already approved and being used in humans today. And this SLB101 capsid is already in use in a clinical trial for DMD by Solid. It's been dosed to several children now safely. So we think the combination of SLB101 capsid as a delivery system and then the MI405 gene therapy product will provide long-term, and I have lifelong with a question mark because as I mentioned at the beginning of my talk, we don't know how long these will really last, but theoretically can produce lifelong protection from DUX4 muscle damage. And we know it can do this in animal models, and we hope that we can get this to humans. And so that's what we're doing now. We're currently fundraising to actually bring this to the clinic. And I want to thank the team. I already mentioned Lindsay Wallace. Some of the highlighted names here are still in the lab. And I also want to highlight Armidus Bio and the team there. This is the biotech company we started that's helping us move this forward. And I will stop there and take questions. Thank you for your attention and time.
Great. Thank you, Dr. Harper. Can you say hello to our audience so we can be sure we can hear you on the chat?
Yeah, hi, everybody. I'm sorry I can't be there.
No, we appreciate you joining. Thanks, Scott. Are there any questions for Dr. Harper in the room?
So if I understood correctly, it sounded like from the muscle injection that you did in the mouse that you have to target the specific muscle itself. How do you intend to deploy it to the human body that's made up of lots of muscles.
Yeah, sorry to clarify that. You're right. The experiment that I described, we did do intramuscular injection in specific muscles so we could use the uninjected muscle as a control, and that way we could measure the effect in the same animal. But some of the work I showed you on the last slide, where we were looking at biomarker knockdown and histological protection, so that's protection of the muscle architecture, and then also the open field, the assay, which is the activity assay, and we've also done some other activity assays and functional assays, including some force measurements that I didn't show on that slide. All of that was done with systemic delivery, so we actually deliver it through the bloodstream, so we can hit a large, basically all the muscles that the AAV capsid is able to hit at the doses that we use. So it's a systemic delivery.
Great. Dr. Harper, we have one question. What are the next steps that will be needed to advance MI405 to the clinic?
Well, right now we've been working on manufacturing, doing test runs to manufacture what would be a clinical grade vector, and then we'll use that to do what's called an IND enabling toxicology study, just to show that this is a safe product in animal models using the manufacturing methods that we're going to require for human testing, and then we're fundraising for clinical trials.
Great. One more question here.
Hi. That was a great presentation. This is Sylvia Blemker from Springbok and UVA. I was curious, have you guys seen how well the treatment penetrates satellite stem cells, and kind of speaking to the longevity of the treatment?
Yeah, we haven't directly looked at satellite cells, but to my knowledge, AAV vectors, including solids, are not known to transduce or infect satellite cells very well, if at all.
Yeah, I'm just curious,
So we're relying on,
in terms of the long term.
Yeah. So that's a really good question about the longevity of this, because the AAV system that we're using, it doesn't actively integrate its DNA. So the DNA that we're delivering, it doesn't actively integrate into the host cell, into the muscle cell of the recipient. So what that means is if that muscle cell gets injured or turns over in some way, it divides, then that DNA that we put in there would be diluted out over time.
Exactly.
So that's a concern about the longevity of it. But the important thing is that muscle, at least, so healthy muscle that's not diseased or damaged, is very long lasting and is considered to be post-mitotic, meaning it doesn't turn over very much, if at all. So what we hope is that, and that's why I mentioned several times in my talk, that we don't really know how long these systems are going to work for muscular dystrophy, for example. That experiment is being done right now. There are people, several hundred people now have been injected with AAV vectors to treat muscle disease. And, we're going to know in the coming years how long these can persist in muscle if they can stabilize the muscle. But if the therapy doesn't work very well, and, let's say DUX4 turns on and we're not able to suppress it well enough, then theoretically those muscles could be damaged and then lose the vector.
Right.
But
Yeah.
now what we're seeing in the mice, the longest we've looked at it in mice is 2 years, and we see stable expression and protection after 2 years in a mouse.
Yeah. It might be interesting to do a muscle injury experiment to see like satellite cell fusion, like how, kind of sort of how would that, the fact that these muscles are going through degeneration, regeneration, how that kind of intersects with the treatment. Obviously, you're preventing the degeneration, but then also, you're also wanting to regenerate muscles at the same time, so it seems like really
Yeah,
interesting to
it's an
sort of mix
interesting
them up.
idea. I would predict that if we intentionally damage the muscle, we would lose expression of our
Right.
therapy.
Great, thank you. I was just thinking, it's like we put Sylvia in as a plant, because we're actually going to talk a little bit later about gene therapies for satellite cells specifically. So unfortunately, we got to move on to the next talk, but thank you again, Dr. Harper, for joining us. Let's give him one more round of applause. Great. So our next presentation comes from Dr. Wes Miller. Dr. Miller is an accomplished leader in cell and gene therapy drug development whose experience includes development roles at Graphite Bio, Astellas Gene Therapies, and Sangamo Therapeutics. Dr. Miller currently serves as Chief Medical Officer of EpiCRISPR Biotechnologies, a company working to develop epigenetic therapies to dynamically control gene expression and treat complex disease.
Hello, my name is Wes Miller. I'm the Chief Medical Officer at EpiCRISPR Biotechnologies, and I would like to thank Friends of FSH for inviting me to give an overview of our EPI-321 program, which is a potential one-time treatment for people living with FSHD. In this brief presentation, I'll hope to cover the following topics. First, review the field's understanding of FSHD as an epigenetic disorder. Then review the design of EPI-321 to address the epigenetic root cause of FSHD. Next, we'll think through some of the unique aspects of a gene therapy approach to treating this disease. We'll look at the summary of some important EPI-321 safety and efficacy or effectiveness findings from the extensive laboratory testing which predated initiating the first in-human clinical trial of EPI-321. We'll do a brief overview of the EPI-321 first-in-human clinical study. And then finally, I'll give my thoughts on a question asked by Friends of FSH, which is what knowledge of FSHD would make developing genetic medicines easier. So first, it is now well accepted that FSHD is an epigenetic disorder. And this abnormality of the epigenome in patients affected by FSHD leads to the abnormal expression of a protein called DUX4 within skeletal muscle cells. Everyone in this audience is well aware that DUX4 protein, when expressed in skeletal muscle, is toxic to those cells. So why is it that people who do not have FSHD don't express DUX4? So as shown in this cartoon here, the DUX4 gene is usually and typically flanked by a very large DNA repeat array called the D4Z4 region. This is comprised of large sequences of DNA units depicted as these triangles on the DNA backbone. What's also known is that many of these DNA units within a healthy D4Z4 are tagged by a chemical group called methyl or methylation. This is depicted by these closed blue circles that are attached to the DNA units. In contrast, relatively fewer of these DNA units lack a methylation or a methyl tag, and this is shown by the open blue circles. In contrast, when we look at a patient affected by FSHD, we of course see a very different story. First, the D4Z4 region is markedly contracted. It is nowhere nearly as long in terms of the numbers of sequences of DNA units comprising it. So this is shown by fewer triangles on this pictogram. In addition to that, however, it's now well described and observed that of the remaining and residual DNA units within the D4Z4, relatively more of them appear to be unmethylated compared to a healthy allele. So this status is called relative hypomethylation. So what's known in human genetics is that genes which are flanked by DNA which is well methylated tend to be silenced. Therefore, at the top in the healthy state, DUX4 is silenced, that is, it is not expressed within healthy skeletal muscle. In contrast, it's well known within human genetics that genes which are flanked by DNA, which tends to be less methylated, are expressed. That is, the gene makes the protein product. And this is why we believe DUX4 is made in patients who have FSHD. So to review, FSHD is caused by, of course, a disease-causing mutation, which is now understood to be less than 10 repeat units in the D4Z4 region near DUX4. This contraction of the D4Z4 is also associated with low methylation on the DNA strand that flanks the gene. Genes in general, which have low methylation, are susceptible to being expressed, that is, the cell making the gene's protein product. It's also helpful to review that sometimes the methylation status of DNA near a gene is referred to as epigenetics. So we can see now that FSHD truly is a problem of the epigenetics or the epigenome. So how is EPI-321 designed to address this epigenetic root cause? As shown at the bottom in this cartoon, EPI-321 comprises three important components. First is something called the guide RNA, which is shown in purple. This is an RNA molecule which is designed to specifically and precisely bind to a region within DNA that flanks the D4Z4. The second component of EPI-321 is a CRISPR-associated protein called dCAS. This protein, shown in the mint green, acts as an anchor. It is attracted to and attaches to the purple guide RNA, as well as the DNA that flanks the guide RNA. Attached to this anchor is the third and very important component of active EPI-321. And these are enzymatic proteins and proteins that are shown in blue spheres attached to the dCAS anchor. These spheres work to remethylate the DNA units that are near the binding site. So as shown in this cartoon, the end result of EPI-321 is expected to be deposition of methyl tags on the DNA units within the D4Z4 that flank DUX4. And by doing that, it is our hope and expectation that we are able to silence DUX4 and copy what it looks like within the healthy state of skeletal muscle. So EPI-321 is a form of gene therapy. As a form of gene therapy, it has two key components. First is the vector. The vector is the component of a gene therapy that carries the DNA gene cargo to the intended tissue. In the case of EPI-321, this intended tissue is skeletal muscle. We are using a vector called adeno-associated viral vector subtype RH74, or more simply, AAV-RH74. This is an engineered viral vector that has now been described to be able to efficiently find, attach, and transduce or egress into skeletal muscle cells to carry the cargo inside. The second component of EPI-321 gene therapy is in fact that active cargo. In actuality, this cargo is simply a long strand of DNA, and this DNA is engineered to encode the three active components of EPI-321 that I reviewed on the previous slide. So these three components are the guide RNA, which is used to find and bind the D4Z4 region, the dCAS protein, which sits down on the guide RNA and acts as an anchor, and then finally the methylator proteins, which work to remethylate the D4Z4 region. As a gene therapy, a single intravenous dose of EPI-321 is expected to be durable, and this expectation is for a few reasons. First, there is emerging clinical as well as large animal experience and data that shows that adeno-associated viral or AAV vectors can and will persist in muscle nuclei for long periods of time. When they persist in this way, the cargo, which is the engineered gene, is continuously expressed. So it is expected that these three active components will be made continuously within muscle cells of treated patients. Secondly, studies conducted by EpiCRISPR with EPI-321's active components, that is dCAS and the methylators, show that even when the engineered gene is purposely removed from a cell system, the methylation marks that were deposited on the DNA when that machinery was there will remain over time, and they are even passed down through generations of cell division. So these two features together are why it's expected that a single dose of EPI-321 could be beneficial and durably so. It's important to consider, though, that in addition to this potentially being a convenient aspect of EPI-321 treatment or in general gene therapy treatment for FSHD, this sort of treatment, which might definitively and continuously silence DUX4 over time, could be crucial to minimize the chance that this protein could leak out within muscle cells. Because every time DUX4 does leak out, we know it has the potential to cause additional and cumulative muscle damage. So prior to EPI-321 being tested in our first in-human clinical trial, it underwent extensive testing in the preclinical setting, that is in the laboratory, to ensure that there was good enough evidence that it appeared effective as well as safe. So I'd like to walk you through some of the key findings of this preclinical experience. First, regarding effectiveness, we estimate, based on carefully conducted FSHD patient sibling studies, that modest differences and increases in methylation near D4Z4 of about 5% might be enough to sufficiently silence DUX4 and prevent symptoms of the disease. In our preclinical experience, we observed that EPI-321 increased methylation above this target in various FSHD patient cell lines which were treated. Similarly, regarding DUX4 and DUX4 pathway expression, EPI-321 suppressed this pathway well over 50% in multiple FSHD patient cell lines tested. Based on the best available animal models of FSHD, it would be predicted that such a deep suppression of DUX4 is likely to translate to clinical benefit. Apoptosis is a medical word and biologic word to describe programmed cell death. Apoptosis is the final pathway after DUX4 toxicity within skeletal muscle. And we were able to observe in the preclinical experience that EPI-321 reduced levels of apoptosis to normal ones upon treatment of multiple FSHD patient cell lines. Finally, we developed models to test FSHD patient-sourced muscle contractility, and of course confirmed that this was abnormal in an untreated state. However, upon treatment with EPI-321, we observed that this contractile force of these three-dimensional FSHD patient-sourced organoids were restored to normal levels. Switching to safety, one of the important focus and features of our preclinical program was to assess in a very careful way for any evidence of so-called off-target gene suppression. This means whether EPI-321 would inadvertently cause the suppression of any other gene except for DUX4 and within muscle tissue. In testing a large number of human cell types, including skeletal muscle, as well as after giving high IV doses of EPI-321 to healthy mice and monkeys, we saw no evidence of this so-called off-target effect. Another potential concern for a product like EPI-321 is whether a protein product that it makes could lead to an injurious immune response. In extensive preclinical testing of EPI-321, including in healthy mice and monkeys followed for over 6 months following dosing, we saw no evidence of this. Finally, very broad safety studies were conducted in mice and monkeys. These animals were followed for up to 6 months following high intravenous dosing, and we observed that EPI-321 was safe at doses of up to 6 times our initial clinical trial dose. So speaking of our initial clinical trial, I'd like to give you a very brief and high-level overview of the design. The study is ongoing and active. It aims to enroll up to 12 participants with genetically confirmed FSHD type 1. The study is multi-center and is being conducted in 3 different countries: the United States, New Zealand, and Australia. Adults who are 18 to 75 years of age at the time of dosing are eligible. Patients are screened, as this is an AAV-RH74 vectorized gene therapy product, to make sure that they don't have pre-existing immune memory to this viral vector. They must be immune negative to proceed with dosing. Patients must be able to take the required immune suppression medicines that are used to minimize the potential for side effects from a viral vector. All participants will undergo extensive vital organ testing to make sure that heart, lung, liver, and kidney appear to be functioning well. As mentioned, this is a one-time administered drug product. However, we are using a staggered dosing approach. This means that before dosing additional patients, we must wait for some amount of time to ensure there are no unanticipated side effects. 2 different dose levels are planned for this clinical trial, 2 times 10 to the 13th vector genomes per kilogram IV, and 4 times 10 to the 13th vector genomes per kilogram IV. And as this is a gene therapy trial, participants will be asked to continue follow-up for up to 5 years following dosing to ensure that there are no late onset safety concerns. Many key assessments will be performed in patients. These include tests of the blood, as well as skeletal muscle biopsies, to understand the biologic activity that EPI-321 might have in patients with FSHD. Clinical or functional endpoints and assessments include strength testing as well as numerous time tasks assessments. Patients will be asked to periodically complete patient-reported outcome questionnaires, which get patients' insight into the nature and severity of their disease over time. And then finally, patients will be required to undergo a serial MR imaging or magnetic resonance imaging to understand if and how EPI-321 might be altering the imaging presentation of their disease. In conclusion, Friends of FSH asked EpiCRISPR, what do you wish we knew about FSHD that would make developing genetic medicines easier? I think it's helpful to think about all of the progress that has occurred in this disease already. First, we now know that DUX4 is the causative protein. This is the target that many people who are trying to develop a treatment are going after. Second, it has been extremely helpful that the epigenetic and genetic underlying cause of FSHD has now been elucidated. It is clearly evident that this is a motivated and organized patient community, and that includes a very well-organized and passionate patient advocacy community. And finally, there are now numerous expert clinical trial sites and clinical trial teams, both in the United States and abroad. In my opinion, the next very important step for FSHD is the identification of an objective and real so-called biomarker of FSHD disease and treatment response.
Great. Let's give Dr. Miller a round of applause, please. Thank you. Okay, Wes, let's test and see if we can hear you.
Hello.
Not yet. We can see you, though.
Let's see. Are you able to hear me now?
Oh, yep. We can hear you, Wes.
Oh, great.
Can you hear me?
Yes.
Wes, can you hear me? Okay, great. I did want to give you an opportunity. I know that since you recorded the video, that EpiCRISPR has put out a press release sharing some news from the ongoing clinical trial. So do you want to give an update to our audience on what was contained in that release and what you guys have learned so far?
Yeah, sure. Happy to do it. So we are continuing to enroll and dose participants in this first-in-human clinical trial. On January 7th, we did a very early and interim data snapshot. 4 participants at that time had been dosed with various follow-up ranging between about a month all the way to about 5 months. Again, this is a gene therapy study that has a design that is common for gene therapies that are first in human, and that is that we stagger patients. And so not all patients can get dosed together. We are very pleased in the initial experience to see that we have really good safety and tolerability. And as mentioned in our press release at the time of the snapshot, we had no serious and no severe adverse event. And so, of course, we will continue to track that closely in the study. We also analyzed some of the strength and functional endpoints for the very early 3-month follow-up visit. But what we were able to assess certainly looked encouraging. They're early data yet. So overall, we were very pleased in the interim data snapshot and analysis, and of course are really excited to see additional patients dosed and additional follow-up.
Great. We had a follow-up question to that one about whether or not the methylation status of these patients has been measured yet or has been changed. Have you seen change in the clinical trial?
Yeah, this is something that we absolutely will be looking at. And so we're collecting muscle biopsy samples before dosing, and then we collect it at various times after dosing. And so we haven't had a chance to analyze those results yet, but absolutely we'll be doing that to understand if within skeletal muscle we are changing the methylation status. Yeah, very, very good question. And absolutely one of the sort of key things we will be looking at from the biosamples collected from participants.
Great. Thank you, Dr. Miller. One question from the chat. Generally, why are clinical trial participants required to be able to walk to qualify to participate in FSHD trials? Is there a possibility for non-ambulatory patients to be able to participate?
Yeah, and I certainly can speak for our thinking about that and kind of rationale for that. I don't know that I could speak for other people who sponsor and design and run clinical trials, although probably their thinking is similar. So a goal of a clinical trial, of course, is to understand whether your investigational treatment is safe and tolerated and also effective at helping patients with the disease. When we look at the best existing natural history data for patients with FSHD, so this means data and information that's been collected for patients who have FSHD but are not receiving any sorts of therapy for it, so in an untreated state. Participants who are in a moderate to moderately severe range with the disease appear to be the ones who, over a relatively short amount of time, will give you or will show that they will progress in a predictable way and in a fairly significant way. And so as you're trying to learn, especially early on, about investigational or candidate treatment for the disease, you kind of want to enroll that sort of population. So for patients who are not ambulant, many of the clinically meaningful and important ways that we understand how FSHD may or may not progress are so-called timed functional assessments. These are things that require participants to do tasks that mimic things we do every day that are important to our quality of life and vitality. And so if someone is not able to perform that timed assessment, then we're not really able to understand if the drug is helping them in that way. Our hope and thinking is that if in our early clinical development and early studies of FE321 that we see the appropriate safety and also benefit profile of the treatment, that in the future we would absolutely want to think critically about either individual separate studies or sub-studies where we enroll patients who are not ambulant and think about how is it that we can test and learn and show whether or not the medicine is helping them.
Thank you, Dr. Miller. Any other questions? I think we could take one more. Got one here.
Thank you. This is very exciting. You mentioned biomarkers and that we don't have an ideal biomarker yet. Two-part question: what will you be tracking, and if the ideal biomarker emerges after your study, will that diminish the utility of what you learn?
Well, I think so. For first of all, your first question, what are we tracking? We are looking at several candidate circulating analytes that could be eventually considered bona fide biomarkers, both of disease severity, but importantly also of therapeutic response for FSHD. And so those are things that we are collecting. For some of these, we are still working on building the assays ourselves so that we can actually measure these particular analytes. But we and others in clinical trials often and frequently collect additional samples, and we ask trial participants at the time of their consent to the study, if when we have leftover samples, if we would be able to use them in order to retrospectively look at, any new, again, analyte from the blood that might emerge down the road as a candidate or even bona fide biomarker. So that's a strategy that we're taking. I know that's a strategy commonly used as well, but hopefully that helps to address that question.
Thank you, Dr. Miller. We're going to move on to the next speaker. Can we have a round of applause for Dr. Miller, please? Appreciate you joining us.
Thanks for having me.
Okay, our next presentation comes from Dr. Peter Jones. He is a professor of pharmacology at the University of Nevada, Reno School of Medicine and serves as the Mick Hitchcock PhD Endowed Chair of Medical Biochemistry. He and his wife serve as co-PIs of the Peter and Takako Jones Lab, which focuses solely on FSHD, including understanding disease pathophysiology, developing new animal models, exploring novel therapeutic approaches, and the development of an epigenetic diagnostic. Their lab has received multiple grants from Friends of FSH Research. Dr. Jones is also the founder and chief scientific officer of Renogenics, a biotechnology company developing the world's first gene therapy to treat FSHD using novel patented CRISPR-I technology.
Hey, this is Peter Jones of the Peter and Takako Jones Lab at the University of Nevada, Reno School of Medicine. Sorry I can't be there. We know this is a fantastic event, the science event and the dinner and raffle and auction that follows. All right, as many of you know, we are a husband-wife team functioning as co-PIs. And one of our many projects, we're exclusively FSHD, but one of them is CRISPR inhibition gene therapy for FSHD. And I should point out first that we also have formed a company where we're co-founders, along with Dr. Carissa Hameda, of a company called Renogenics, which is a spin-out of UNR. And the purpose of this company is to provide a platform to bring our CRISPR inhibition gene therapy to clinic. Okay. All right, so I don't need to remind everybody, but I will anyway, that all forms of FSHD are caused by a pathogenic gain of expression of the DUX4 gene in skeletal muscle. And this was, DUX4 originally described by Alexander Belyaev, but the real key work showing that it was truly the pathogenic gene in FSHD was produced by collaboration between Silver Vandermeer's lab, Richard Lemmers, Ravi Twill, Stephen Tapscott, Dan Miller, many people up in the Seattle area that Friends of FSH has funded, right? And so they let us know that the key therapeutic target is the DUX4 gene and either the destruction or prevention of the DUX4 mRNA, okay? So when a cell expresses DUX4, a somatic cell like a muscle cell in FSHD, many things happen. Okay, not sure which of these, if any, actually cause FSHD. Maybe multiple ones cause FSHD. But one thing seems very clear, and that is that if you can destroy the DUX4 mRNA for something like antisense technology, such as Avidity, Arrowhead, Myricule, Dyne, Souffle, all of these companies that have special molecules that can destroy the DUX4 mRNA, that should have therapeutic benefit. Okay, you could destroy the RNA with a CRISPR-Cas13 approach, although that's not working very well. Or you can prevent as we're doing, prevent the expression of the DUX4 mRNA by silencing the DUX4 gene. Okay, and we're going to do that as a CRISPR inhibition technology. Now you could use a different CRISPR to do something somewhat similar. Not actual traditional CRISPR editing, but CRISPR base editing. This is work from Silver Vandermeer's lab that's kind of a little bit further behind, not quite ready for the clinic, but that these have some viable potential. All right, so CRISPR inhibition. So CRISPR, you think about it as essentially very specific genome targeting technology, okay? Cas9, kind of the CRISPR machine, traditionally cuts DNA, okay? But it cuts DNA very specifically where a guide RNA tells it to go. What we're going to use is something called dead Cas9, where we've eliminated the cutting ability, but maintained the genome targeting specificity. And now we're going to fuse an activity to this. And the activity we're going to fuse is either a transcription repressor or an epigenetic chromatin modifier, and use this to shut off the DUX4 gene in a very specific way. Okay, this is CRISPR inhibition, different type of CRISPR. Okay, we all know, right, that FSHD is caused by epigenetic dysregulation at the 4q35d4z4, which leads to an aberrant activation of the DUX4 gene from within the array, right? A healthy sized D4Z4 array greater than 10 or 12, 15 repeat units, up to 100 repeat units, is epigenetically silenced, and the DUX4 gene, which is present in each one of these triangles, is off. Okay, it's also a similar array in chromosome 10. These are all epigenetically silenced, and in individuals that do not have FSHD, 2 of these arrays typically are both silenced. However, in FSHD, the contraction leads to this epigenetic disruption, okay, and therefore the DUX4 gene located in the very last repeat unit is able to be expressed. And this is how you get expression of DUX4 mRNA, DUX4 protein, and the initiation of pathogenic cascade. Okay. So now if you're going to use CRISPR genome targeting technology, very, very specific genome targeting, and target the DUX4 gene, the thing to remember is that all of these D4Z4 units have that sequence. So you have so the problem is not a non-specific problem. The problem is a specific problem, right? There are many specific targets in FSHD, and this is unique to the repeat genome. Okay, so if you're going to cut the DUX4 gene in traditional CRISPR-Cas editing, all of these are going to get cut, and that's going to be a disaster for the cell. It's actually going to lead to cell death. Okay, and that's basically would speed up FSHD or muscle cell destruction. All right. However, CRISPR inhibition is going to turn off the repeats without doing any damage to the DNA. Right, we're going to drop an off switch right here on the pathogenic gene that's being expressed, turn off, epigenetically turn off, or just transcriptionally repress the region. Okay, all the other genomic loci are already off. Okay, so instead of getting cut, they're all going to be turned off. Now, in theory, this will work really well, right? But also, that's an awful lot of off switches and a lot of the CRISPR. And so nobody had ever shown that you could do this in FSHD or actually any repetitive sequence in the genome, right? Normally people work on what are called single copy genes, and those are much easier to specifically target than the repeat genome, because this could actually be a huge sponge or a sink of CRISPR, and maybe it wouldn't work. Well, it turns out in 2015, Carice Hameda in our group, who came from Steve Hauschka's lab at the University of Washington, where she got her PhD, probably the best place you could possibly get your PhD if you're interested in muscle gene regulation and skeletal muscle cell biology. And she in our lab showed that this can in fact work for FSHD. CRISPR-Cas9 transcriptional inhibition, right? CRISPR inhibition can silence the DUX4 gene. The first time ever targeting the repeat genome, and actually we got a lot of press on this because it was the first time CRISPR had been used therapeutically outside of cancer. We actually beat the Duchenne people by about 6 months. All right, now, right, it's 2015, it's 2026 now. So we've spent the past 10 years going from the lab to the clinic, because we cure a lot of things in mice, we cure a lot of things in the lab. You guys all know that, right? And, but curing something in people is very different, because taking a therapeutic to the clinic is much more difficult. So we've been building a clinically relevant CRISPR inhibition machine for FSHD. And what that means, we have to get everything functionally into a single delivery platform, and we need to use adeno-associated virus, or AAV, okay? So the requirements for the activity that needs to go into the virus, we need to be able to silence the DUX4 gene in FSHD1 and 2, expression in skeletal muscle, little to no expression in unaffected tissues. That's actually very, very important. And it has to all be small enough that all the components, including the guidance system, the Cas9 repressor, fit into the AAV vector for delivery. Okay? So we have done that. There's a size restriction of 4,400 bases of DNA, the payload for AAV, right? And we put in the gene expression regulation to tell it where it goes, the CRISPR-Cas in the machine, the effector, and the guide RNA. Now, EpiCRISPR Biotech has done this as well, except they've done it a different way. They've made a very small Cas9, dead Cas9, and effector What we've done is we've made mini regulatory cassette and a mini repressor. So different ways to get at the same thing, which is to fit a CRISPR inhibition system into a single AAV. Okay, so as we mentioned, the first generation was too big. We had a very large regulatory cassette, a very large Cas9 protein, and the guidance system, the guide RNA, had to be in a separate virus, so we had to use two viruses. So fortunately, as I mentioned, Carice had got her PhD and did some postdoctoral work in the Hauschka lab at UW, and she's one of the world experts on the skeletal muscle, or striated muscle, I really should say, on gene regulation and the regulatory cassettes used in current muscular dystrophy gene therapy trials. And so she looked at the sequences and said, I can make a much smaller one and better. One of the keys is that in FSHD, your heart really doesn't need therapeutics. It's not really pathologically affected. And so she was able to remove the heart regulation and move everything together. It's not random, but very carefully and specifically arrange the regulatory elements into a much tighter, smaller cassette and maintain activity and save us a lot of space so that we could actually fit everything in, right? And one of the keys is that she actually maintained the triple activity of the promoter. Okay, and we can show you this by what we did is we took her FSHD optimized regulatory cassettes. The one we like the best is called Have a Little Heart, and we fused it to a fluorescent red reporter called Cherry, and we injected this in a virus into mice. It said, where is this expressed? Everything places expressed will glow red. Okay, so if you look at the TA, very red. Gastroc, very red. Soleus, EDL, quadriceps, very red. Back, diaphragm, and a little bit in the heart. Now, if you look in the gut and the liver, and actually I could show you many other tissues, you see no expression. Okay, so this tells you it's very high level of expression in skeletal muscle, very low in heart, much lower than the original cassettes, and virtually no expression in non-skeletal muscle tissue. So fantastic for FSHD. You might ask, well, was the virus there? And actually, it turns out we use RH74 serotype of AAV, and this goes everywhere. It's a horrible virus, actually, to use. A lot of it goes to the liver, a lot of it goes to the heart, a lot of it goes to the kidney. But as we showed you, the gene expression is not there. Okay, and so this is very specific gene expression. All right, so we were now able to take our HLH, Have a Little Heart promoter. We also used a smaller version of dead Cas, and then she had to make I'm not going to show you the evidence, but she then had to engineer and create smaller epigenetic regulators, which we did and we recently published. And basically, she was able to get everything to fit into a single virus, and it works spectacularly. I'll show you that here. We took our all-in-one CRISPR system and we put it into FSHD1 myocytes, and you can see we're getting 70, 80% repression of DUX4 and DUX4 target genes without adversely affecting myogenesis. And FSHD2, it works even better. Okay, so our all-in-one system clearly represses DUX4 transcription and target genes. If we look at the general, DUX4 itself is a transcriptional regulator, so DUX4 induces many genes aberrantly, and this causes problems in cells. And what we show here in red, genes that were up in FSHD are now down when we treat them with our all-in-one CRISPR inhibition. And in green here, these are genes that were repressed by DUX4 are now up-regulated. In the end, we have returned the FSHD transcriptome to a healthy state. Okay, it's also done the same thing in FSHD2. And you can see out here, there are very little colored dots outside. This is very specific. Okay, so next important thing is to pick the right virus. You want to get to clinic, it's not just important to have a great cargo, you need to have a great delivery system. And AAVs, adeno-associated virus, these next generation viruses are being developed to transduce skeletal muscle with very low immunogenicity, low tropism beyond skeletal muscle. This is very important because it will reduce viral load, reduce cost, reduce liver toxicity, and increase safety. Okay. So we're going to do this now. We're going to go into a mouse first with our next-gen virus, and we have generated the FluxDux4 FSHD-like mouse in the lab. Takako Jones did this, and virtually every therapeutic you're going to hear about goes through the FluxDux4 FSHD-like mouse model. This is what these little guys look like. basically they're tamoxifen-inducible pathology. And you can see this guy's just having trouble getting around while his siblings that are not expressing DUX4 are having a good old time running around, right? Maybe you feel like this guy sometimes, right? So this is our mouse model, double transgenic. When we put in our CRISPR all-in-one with a next-generation vector, we're able to maintain weight after tamoxifen injection and actually increase treadmill running, so we get functional improvement while DUX4 target genes are decreased. Okay. Next question is, will this therapy actually work in an FSHD patient? To do that, we went to our humanized xenograft mouse model where we took cells from an FSHD patient, immortalized them, and then grew an FSHD human skeletal muscle in a mouse. And it's very important how you do this because you can do this incorrectly and get a bogus result, quite honestly. But what we do is we grow an FSHD muscle that is innervated and vascularized and can contract first, and then we systemically treat the mouse with our CRISPR inhibition all-in-one, and we show whether or not we can get benefit. And what you can see here with our non-targeting versus our targeting, we get wonderful repression of DUX4 in an FSHD, human FSHD muscle grown in the mouse. Okay, finally, what we wanted to do is we know a lot of mouse studies don't translate to humans. And so with the help of Friends of FSH Research, we have generated FSHD-like mini pigs for testing FSHD therapeutics. The general idea is, it's great to cure stuff in the lab and cure stuff in mice, but going from a mouse to a human can actually be very, very different for delivery, specificity, dosing, efficacy, and durability. Okay, so we need a larger model. On other diseases, how to use dogs. We use pigs. We created, again, Takako Jones in our lab created these DUX4 flex tamoxifen-inducible mini pigs, and you can see here that their muscle looks very much like an FSHD muscle. We get a lot of fat and down here in red and fibrosis here in white. And this is going to be very important because a viral delivery or delivery of any therapeutic has to get through this, because this is what you look like. Getting through this is much easier. So delivery and dealing with this toxic environment is very important. So we take our all-in-one, and we want to basically, the idea is to take take our virus for the next-gen load. We started in patient-derived muscles, moved to a flexed mouse model, moved to a humanized mouse model, and now through a large animal. And this is where we are now, finishing up our large animal studies, and to take only the best therapeutic into the clinic to treat patients. Okay, so I want to thank everybody for this. Is actually, here we are at one of the Friends. It's the same event that you're at now, several years ago with some of our good friends, and many other events. We always want to remind everybody who we're working for and why we're doing it. So thank you to all the patients who are involved, and thank you to our funders and the Friends of FSH Research in particular for helping us out. We got a great team here in Reno, and hope you guys come by and see us sometime. Happy to take any questions.
Great. Thank you, Dr. Jones, who's joining us live. Any questions from the audience? We have one in the chat. Okay. One question from the chat is about kind of capacity for the technologies that we use for delivering. So could you use 2 AAV capsids to have more room to fit a larger and stronger methylator? Could you use a lipid nanoparticle with a larger capacity? How do you think about sort of balancing? You talked a lot about reducing the size of the transgenes. How do you think about balancing that with the efficacy of the molecules that you're developing?
Okay, I guess there's 2 answers to that. One is we're quite happy with the efficacy of our repression. So we don't feel we need to go any smaller or bigger, I guess is what you would say. But the general question, one of the great things about the next-gen vectors, and this could apply to FSHD, it could apply to DMD and other diseases, is that because you can use such a much lower dose, you could actually use multiple AAVs. It's kind of if you, and I know Jeff Chamberlain has talked about this in the past at other events, that this is potentially where it's going with the incredible efficacy on low doses of the next-gen vectors. Lipid nanoparticles, that's a possibility. I would imagine at some point non-viral delivery is where gene therapy is going. You got to get to targeting, you got to get to biodistribution and everything. I think that's just a technological limitation right now. Right now, where we are, AAV is your best delivery. And if you can fit it into one, I still think that's the best way to go. And especially with the next gen vector. And really, when it comes to CRISPR inhibition, ApiCRISPR has also shown. We can get enough repression into one vector that we don't need. Now, if you wanted to do like DNA editing, CRISPR editing of a sequence, those are much larger, and potentially you could. I suppose you could do some sort of fusion, but I'm a bigger fan of the technology always driving to get smaller stuff and better stuff. People are always. It's amazing that what goes on in the background. There's always a new thing coming out that's going to be better. So.
Thanks, Dr. Jones. This might be the first that this audience is hearing about the mini pig model. You described some of the benefits of it. Can you remind us again what can be learned from the mini pigs that you think maybe is not being modeled as well in the mouse model, and what have you guys learned so far?
Sure, fantastic. Well, so the mini pig. So one thing about a mouse and a human genome in respect to DUX4 are very different. The mouse, if you put human DUX4 into a mouse, you induce 6,000 genes, just like if you put human DUX4 into a human. But they're virtually all different genes. The transcriptome is extremely different. But in the pig, when we worked on, Stephen Tapscott did this work with us up at the Hutch, and showed that when you put human DUX4 into the pig, the transcriptome, the same target genes are activated, the same retrotransposons are activated. So it's like a very human-like transcriptome. So that means we can understand better pathology, human-like pathology downstream of DUX4. Pig has a very human-like immune system, and I don't think it's appreciated necessarily how important the immune system is going to be in FSHD and also in gene therapy necessarily. There's a reason people use dogs and pigs, not just because you want a large animal model, but you need to replicate more human-like immune response, because this is involved in all the neuromuscular diseases. So we get an amazing FSHD-like phenotype with very small amounts of DUX4 expression in the pigs. Again, mice are very lean. They're not particularly FSHD. We call them FSHD-like. And I think this is super important for something that's beyond what we're doing, but the next step for everybody in the audience is building your muscle back. Once we stop the destruction, it doesn't matter if it's FSHD for us or Avidity or Dyne or whoever, you're going to stop the destruction. You want to get your muscle back. And the pig really replicates, we can actually essentially give it the pathology and then stop the destruction, mimicking a therapeutic because of the way we designed it. And now you have a fatty, fibrotic FSHD large animal, which is you, and we can test things for how to get muscle back. Stem cell, whether it's cell therapy or myostatin inhibition. There's a million things that people are trying to get muscle back. And I think that's going to be really the benefit of the pig is going to be for the build it back. And then the last thing on the pig that's really important is biodistribution of gene therapy or biodistribution of biodistribution of anything. A healthy monkey is not FSHD. A mouse is not. So actually understanding how your therapeutic, whatever it is, distributes through a fatty, fibrotic muscle like you is actually really important. So we're learning a ton from the pig on immunology, biodistribution, dosing, and I think it has saved us from having a failed clinical trial, quite honestly. So I'd rather go slower and have success than go quickly and make a mistake that could have been avoided.
Excellent.
And thank you
so
for
much,
Friends
Mr.
for
Jones.
funding that, by the way. Thank you guys for funding part of that.
Thank you, Dr. Jones. Let's give him a round of applause. Appreciate your time. We had
Sorry I
some
can't be there.
bristling in
Hope
the
you guys have a
audience
good
about
time.
calling people a fatty, fibrotic muscle, but I think we'll get over it. Thank you, sir. We appreciate it.
We're all pig-like.
Great.
That's all
Our
thank you.
next presentation comes from Dr. Wendy Agnesi, who currently serves as Senior Global Medical Director, FSHD at Avidity Biosciences. Dr. Agnesi is an accomplished industry leader who has spent her career in various leadership roles in the pharmaceutical industry. Most recently, before joining Avidity, she was head of the U.S. medical team for rare disease at Biogen. She is passionate about drug development and advancing what is possible for the rare neuromuscular communities that she serves. So thanks for joining us, Dr. Agnesi.
Thank you to the Friends of FSH meeting organizers for the opportunity to speak with you today on an update from Fortitude, our randomized phase 1/2 trial studying Delbrax in adults with FSHD. A few disclaimers before we jump into the presentation content. Please take a moment to read through these. This presentation discusses the clinical development program for an investigational product, Delbrax. No conclusions regarding the efficacy or safety can be drawn from these initial data or presentations, as Delbrax still is investigational and the product has not been approved by any regulatory agencies. Its safety and efficacy have not been established, therefore this is truly just for your educational purposes only. On behalf of the Avidity team and personally from someone who's worked in the neuromuscular space for almost 2 decades, I are grateful to the network and community of stakeholders that have paved the way, dedicated their time, knowledge, resource that's helped inform and advance our Fortitude clinical development program. This includes the FSHD Clinical Trial Research Network, the natural history studies and registries, all the wonderful patient advocacy groups, and of course, the patients, families, and caregivers. So let's get into it. We will be discussing the Fortitude Delbrax phase 1/2 clinical program. First, let's level set on what Delbrax is. It is an investigational antibody oligonucleotide conjugate, or AOC for short, formerly referred to as AOC 1020. And just very briefly, there are 3 colors on this cartoon figure, as the AOC has 3 parts to the molecule. The blue part is the antibody, and that targets the receptor on the muscle cell, and I'll show you this on the next slide. The green part is the linker. This enhances the stability of the molecule and helps maintain its structural integrity. And the red part is the oligonucleotide part, which are designed to precisely engage with the RNA target. And this construct, or the AOC platform, is designed specifically to deliver RNA therapy to the muscle. So let's take a deeper look at this. So in the upper left, you're seeing that 3-color AOC again, which is Delbrax. The monoclonal antibody portion of the AOC, that blue part, is designed to target a specific receptor on the muscle cell. In this case, it's the TFR1 receptor. This allows that red oglio part, that section, to get into the muscle cell and bind to their RNA targets and modulate or modify or change that gene expression. And in the case of Delbrax, it's targeting the DUX4 gene to turn it off, so it does not make that DUX4 protein, which is known to be the root cause of FSHD. So with that high-level background on Delbrax, let's discuss how it was studied in the ongoing clinical development program. So here's an overview of the Fortitude study design. This is a 12-month phase 1/2 dose escalation study. And if we focus on the bottom half of the slide first, it's a randomized, double-blind, placebo-controlled study, which included participants ages 18 to 65. And the primary and secondary objectives focused on safety and tolerability, as well as pharmacokinetics, PK. Anything related to the efficacy, the functional outcomes, the biomarker endpoints are all exploratory in nature. Okay, let's go to the top half of the slide. Participants were randomized to receive either Delbrax or placebo. The 2 horizontal rows in the middle, the top blue row represents part A of the study, which is the 2 milligram per kilogram dosing arm, and the darker blue in the middle is part B, the 4 milligram per kilogram dosing arm. And within those 2 rows, those little images or the IV VAG is where participants received their dose of either Delbrax or placebo, depending on which arm they were randomized to. And after patients or participants finish the 12-month randomized placebo-controlled portion in this study, they can enroll in the open label extension. And at that time, all patients receive Delbrax 2 milligrams per kilograms at a regimen of every 6-week dosing. Here we are showing the baseline demographics and characteristics of the participants in the study, with the 3 columns representing those participants who were either in the placebo arm, the 2 milligram per kilogram arm, or the 4 milligram per kilogram arm. And you can see the respective number of participants in each of those groups. You can see listed out, 92% of the people in the placebo arm were male, while 62% and 50% were male in the Delbrax 2 milligram and 4 milligram per kilogram arms, respectively. The other characteristics were fairly well matched across the groups, with all participants being genetically diagnosed with FSHD1, despite the fact that the study protocol allowed for both FSHD1 and FSHD2 participants in the study. And to call your attention to the FSHD clinical score, for background, the FSHD clinical scores are range between 0 to 15. 0 is no sign of muscle weakness is present, whereas 15 equates to all muscle groups that were tested or severely impaired. So the mean here hovers around 9, so it's kind of right in the middle of that clinical range. Looking at the long-term safety and tolerability, and what's reported here is that 12-month top-line data time point from Fortitude, along with the 38 patients that enrolled into the open label extension at the time of this analysis. All patients remain enrolled in the study at the time of this analysis. There were no discontinuations. Most AEs were mild or moderate. No related severe or serious AEs to Delbrax. Most common related AEs occurring in greater than 3 participants included fatigue and hemoglobin decreases. There was 1 unrelated severe non-serious AE of herpes zoster, which occurred in 1 participant, and 3 unrelated severe serious AEs of fractures in 1 patient. And before I show the biomarker results, a quick comment on biomarkers in general. A biomarker or biological marker is a measure or indicator of some biological state or condition. They can be used to examine normal biological processes, pathogenic processes, meaning something that's causing a disease, or used to measure pharmacological or drug responses. And unlike muscle biopsies that are kind of a sampling of that single muscle and sometimes considered fairly invasive because that's how the samples are collected, and that's that image on the left, a circulating biomarker is minimally invasive and provides an opportunity for early detection of whole body response to a treatment, in this case, Delbrax. So let's get into the results. This is exploratory analysis of the novel biomarker KHD-C1L or CDUX. You'll be hearing me use those interchangeably. CDUX is directly regulated by DUX4, and we commented on that earlier slide that DUX4 is known to be the root cause of FSHD. And if you look at the figure on the right, this is showing that compared to healthy controls in both the Resolve data set and the Fortitude clinical trial data set, CDUX, or that KHD-C1L, is elevated 6 to 9-fold in people living with FSHD. And now looking at the Fortitude biomarker data, we see a statistically significant difference in CDUX or KHD-C1L comparing placebo, which is that gray line, compared to Delbrax, the orange line, over the course of the 12-month study. And just reading the title summarizes it very well. A rapid and significant decrease in CDUX followed Delbrax treatment. These results helped inform the dosing schedule for future subsequent Delbrax trials, which we'll touch on in a few moments. So let's move on to the exploratory efficacy or functional endpoints from the trial. And when I say the word exploratory here, it means that while these data are directional and formative, particularly to inform clinical trial design moving forward, this does not establish the drug efficacy or effect. As I previously mentioned, for this study in particular, the primary endpoint was safety. I'm going to show 4 slides that all have the same formatting or look to them. So we'll take an extra moment here on this first one just to ensure that we are all oriented to the key components of the slide. We mentioned that these are exploratory in nature, and what we're reporting on here is data at 12 months, which utilize the every 13-week dosing regimen. The cartoon on the left is a visual to help describe the specific test we're speaking about. Here it's the 10-meter walk-run test, which is the time it takes to go 10 meters or about 30 feet as fast as the participant can do safely. And then let's move to the figure on the right. The x-axis, or the horizontal line with the 0, 1, 2, 3, 4 seconds indicated on it, is going to be that change from baseline over the 12 months. The vertical dotted line at the 0 point would represent if there was no change over this time period. And then the orange bar is Delbrax and the gray bar is placebo. So what we're showing here is the improved functional performance on the 10-meter walk-run test for Delbrax treated patients compared to the placebo group. Here we're looking at the same setup for the slide, but reporting on the mobility measure, the timed up and go, or TUG. This is traditionally a measure of balance. It's the time it takes for someone to get up from a chair, walk with their casual gait for 3 meters, then they turn around 180 degrees and walk back to the chair and sit down. These results show an improved performance or that functional mobility on the timed up and go for participants in the Delbrax treated group compared to the placebo group. Let's now look at another exploratory endpoint, which is QMT, or quantitative myometry, which is a measure of strength. This is measured with a handheld device to isolate different motor tasks, and here we're looking at specifically the elbow, the knee for flexion and extension, the ankle for dorsiflexion, and the shoulders for abduction and rotation. These measurements collectively then create what's known as a composite score, and is reported here as QMT percent predicted normal score. You can see that there was an improved strength in the Delbrax treated patient group compared to the placebo group. Here is the last of those 4 slides that I mentioned, and this is the reachable workspace, which focuses on the function of the upper extremities to detail the volume of the space that someone could reach into in different quadrants, is illustrated by the cartoon on the left. On the right, we are showing the average of the quadrants 1 and 3, and the Delbrax treated patients improved in their reachable workspace compared to the placebo patients who are decreasing in their reachable workspace over that 12-month period. These next 2 slides are also exploratory analyses and focus on patient-reported outcomes. We're showing that change again from baseline at 12 months. The vertical dotted line represents no change during the 12-month study, and improvements on these outcomes is to the right, as the green arrow is illustrating here. Delbrax treated patients are in the orange and placebo in the gray again. And as reported on the PROMIS assessment tool, you can see improvements in physical function and fatigue for patients treated with Delbrax compared to the placebo group. And similarly, on the NeuroQual, looking at the upper extremity score as well as sleep disturbance, you can see a similar directional trend. Lastly, looking at pain and overall health, as well as the patient global impression score, we see improved quality of life in Delbrax treated patients compared to placebo, as reported here. So what we discussed so far was the completed top-line data from the Fortitude Study Clinical Development Program Parts A and B. There are also some ongoing trials with Delbrax, and want to touch on those briefly here. The Fortitude study has a Part C, also known as the biomarker cohort. This is also a randomized, double-blind, placebo-controlled study, which will enroll 51 participants. If you recall from an earlier slide, I mentioned that the learnings from earlier on in the Fortitude program helped inform future trials. Well, this is one of those trials where we're now using the dosing regimen of Delbrax 2 milligrams per kilogram every 6 weeks. You can see listed a few of the key considerations for the study design, including that participants of ages 16 to 70 will be included in the trial. And the primary endpoint is the change in that circulating biomarker KHD-C1L, also known as CDUX. This is a 12-month study, and we anticipate the top-line data readout from this study in Q2 of next year, 2026. Also ongoing is our Fortitude 3 global confirmatory study. This will use that same dosing regimen as the biomarker Part C study, the 2 milligram per kilogram every 6 weeks, but this will be 200 participants, and the length of the trial is 18 months. This will also be including both FSHD1 and FSHD2 patients, and the age range here is 16 to 70 years of age as well. We'll have approximately 45 sites in North America, Europe, and Japan, and the efficacy endpoints, as noted on the slide, will include the 10-meter walk-run, TUG, and QMT. In summary, our clinical development program is on track to be the first disease-modifying therapy in FSHD. We demonstrated a reduction in that KHD-C1L or CDUX, a DUX4-regulated circulating biomarker. The exploratory analysis of the 12-month data showed improvement for Delbrax-treated patients compared to placebo across a number of different functional endpoints, as well as the PROs we mentioned. And we are rapidly advancing the Fortitude biomarker trial, as well as that global confirmatory phase 3 study. We've aligned with the FDA on accelerated approval, as well as full approval pathways for Delbrax, and initiated the studies accordingly. I mentioned the biomarker C readout is anticipated in Q2 2026, and the phase 3 or Fortitude 3 readout and global regulatory submissions are expected in 2028. With that, I want to sincerely thank all of the study participants, the families, the investigators, the research team. This is definitely a community effort to advance what is possible in FSHD. And with that, I thank you for your attention.
Great. Thank you so much, Dr. Agnesi. So we have a couple of questions for you here. We heard some exciting news in the fall that Novartis intends to acquire Avidity. Are there any updates, and what does that mean for the FSHD Delbrax program?
Yeah, first of all, thank you again. Sorry I couldn't be there with you in person. So regarding the announcement, there was a press release back in October of last year, and it was announced that Novartis plans to acquire Avidity, and that closing of the acquisition is anticipated in the first half of this year. We will work closely with the Novartis team to ensure that the registrational studies that we just touched on continue to run smoothly after the transaction closes, and that we're committed to supporting that seamless transition for the programs. When we think about Novartis, they are one of the world's leading global health companies, and they're very well positioned to continue to advance the Delbrax programs through commercialization. And we're confident that this transaction will support the global expansion of the neuroscience pipeline that we have within Avidity at this time. I do want to reiterate maybe some of the points from that summary slide that we had as far as next steps for the program. From a regulatory perspective, we do have that alignment with FDA that we have both the accelerated approval pathway and full approval pathways open to us as potential scenarios moving forward. That global Fortitude 3 study that we've initiated is intended to support the global approval for Delbrax, so that continues to be a priority for the team. And that data readout, as well as regulatory submissions globally, are expected in 2028. So those milestones continue to hold true as we think about next steps with the acquisition.
Great. Thank you, Dr. Agnesi. What are your plans for pediatric patients for Delbrax?
Yeah, thank you for that question. So the studies that I showed so far, these are the adult studies. We absolutely understand the enduring need for treatments across the spectrum of ages in FSHD. So we are committed to advancing that phase 3 program, as I alluded to. The Fortitude study data that I showed you, that inclusion criteria was in adults. We've taken those learnings and expanded the patient population for cohort C, as well as the phase 3 program moving forward, where we will have some adolescent patients in their ability to enroll. So that age bracket is now 16 to 70. So that is now ongoing. Those enrollment efforts for the phase 3 program. We are working with stakeholders within the community to really understand what the needs are going to be for a study protocol for the pediatric program, to make sure that we have a study that will help support the commercial access of Delbrax for the pediatric patient population moving forward. So those are ongoing discussions now, and we are absolutely committed to making sure as we reach milestones and decision points in that process, that we will come back and share more information regarding pediatric programs moving forward to the community at large.
Great, thank you. And one last question, what should a patient do if they want to participate in the Fortitude phase 3 study?
Yeah, thank you for that question regarding clinical trial participation. And and I say we collectively, whether it be pharmaceutical companies, biotech companies, academic researchers, we would not be able to advance the research that we have available to us if patients, families were not willing to participate their time and participate in the research. So thank you for that. Regarding the Fortitude Phase 3 program right now, the study sites have been selected so far. So if you're a person living with FSHD and might be interested in the trial, I would advise that you visit the study website. So if you just type in Google Fortitude study, it'll take you to the website where it has a number of different resources and information there. One of the tabs on that website is a locations tab, and it will have all the study sites that are participating in the study. And within that, they're designated who is still open to screening. So that's a good first stop for you as far as understanding what sites might be geographically feasible for you. Another step in the process is to make sure that you're having that conversation with your healthcare provider to ensure that this particular study, based on the inclusion and exclusion criteria and our understanding of Delbrax to date, that this would be a feasible study for you to participate in. I do want to mention related to that, we did get tremendous interest in the study itself. And because this study opened last year, there are a number of wait lists already at a number of the sites that we have. So I would, again, if it's something you're interested in, have that conversation with your healthcare provider and understand where the sites are that might still be screening patients for the study.
Great. Thank you so much, Dr. Agnesi. Let's give her another round of applause.
Thank you.
Okay, our last talk today is from Dr. Ellen Padilla, postdoctoral researcher in the Chamberlain Lab at the University of Washington, focused on gene therapies for muscular dystrophies. Dr. Padilla completed her PhD training in the lab of Dr. Stephen Tapscott at the Fred Hutch, where she focused on studying genetic mechanisms of FSHD pathogenesis and the consequences of early embryonic gene expression in cancer. She also received funding to support some of the work that she will present today from her Friends of FSH research.
My name is Ellen Padilla, and I'm currently a postdoctoral researcher in Jeff Chamberlain's lab at the University of Washington. As a PhD student in Stephen Tapscott's lab at Fred Hutch, I was honored to receive funding from Friends of FSH, the Chris Carino Foundation, and the Seattle Wellstone. Today I'm going to outline that dissertation work as well as share some of my goals for my postdoctoral work as an early career scientist in the muscular dystrophy field. As you all know, FSHD is caused by a loss of epigenetic repression of the DUX4 gene. Loss of DNA methylation and relaxation of chromatin allows for expression of this early embryonic transcription factor in skeletal muscle. This misexpression of DUX4 and its transcriptional program can lead to increased oxidative stress, inflammation, DNA damage, and other aberrant cell effects, leading to chronic muscle wasting and dysfunction and a muscular dystrophy disease progression. The genomic landscape of the DUX4 gene is complex, with coding transcripts located within each D4Z4 unit in a D4Z4 macrosatellite array. Unaffected individuals typically have between 11 and 100 D4Z4 units, leading to strong transcriptional repression. A reduction in D4Z4 copy number or mutations in epigenetic repressors can lead to FSHD disease progression. Ultimately, FSHD forms a complex disease continuum with highly variable numbers of D4Z4 repeats and mutations in epigenetic regulators, both contributing to pathogenesis. Previous studies attempting to elucidate mechanisms of D4Z4 regulation have shown that many different signaling pathways, histone modulators, DNA methyltransferases, and other epigenetic repressors have been identified as regulators of the D4Z4 and repressors of DUX4 expression. The complex nature of all of these different interacting factors in one region has made determination of essential factors for FSHD therapeutic intervention very difficult. Mechanisms of recruitment of these factors, what initiates silencing, and what maintains this silencing are all still areas of active study. The goal of my project was to determine if there were specific D4Z4 sequences that were responsible for these mechanisms of repression. To address this question, I developed a reporter assay that should assess the ability of any sequence to recruit silencing machinery. We can take any sequence of interest, so D4Z4 sequences, and insert them upstream of a constitutive promoter driving GFP. This is then knocked into the AAVS1 locus, which has low intrinsic silencing activity, into cells. And so if a sequence does not recruit repressors, there should be high GFP expression from the construct. If, however, the sequence can recruit silencing machinery, GFP expression will be suppressed. This leads us to a simple readout for looking at the silencing activity of a specific sequence of interest. To test the validity of the system with a disease-relevant sequence, the LRIF1 promoter region was inserted into the reporter construct. SMCHD1, a FSHD2 modifier, is known to bind and silence this region. This LRIF1 promoter sequence was shown to decrease GFP fluorescence when compared to a neutral control sequence, as you can see by this shift in the peak towards the left here, towards lower GFP fluorescence. This indicates that our silencing assay works. When applying this assay to D4Z4 sequences, there were several possible outcomes that we proposed based on previous research. The first is that the repetitive nature of the D4Z4 with multiple copies of this 3.3 kilobase repeat in tandem leads to silencing. This is supported by the fact that many repeat arrays throughout the genome are silenced, and that in FSHD, repeat copy number is directly associated with disease severity. So if this is the main driver of silencing, no individual D4Z4 region will silence the reporter construct. The second model is that epigenetic repression is distributed over each individual D4Z4 repeat. The D4Z4 is highly GC rich and has a high density of these CpG sequences that can be methylated and silenced. In somatic cells, the D4Z4 is also highly methylated over the entire region. If this is the main driver of silencing, all D4Z4 regions will silence the reporter system. Finally, the third model for D4Z4 epigenetic repression is that a kill switch sequence is first modified and then drives further repression of the rest of the D4Z4. This important regulatory sequence would specifically recruit silencing machinery, which in turn promotes silencing for the rest of the region. In this case, only specific D4Z4 sequences will silence our reporter construct. So, to test this, one D4Z4 unit was divided into 14 overlapping fragments. These sequences were cloned into the reporter construct before knock-in to this AAVS1 locus in HeLa cells. Flow cytometry of GFP showed no silencing activity in most segments. GFP fluorescence peaks shifted towards the left indicate repression of GFP. However, as you can see, surprisingly, one segment, named D4Z4 segment 5, was sufficient to suppress reporter expression, indicating that this third model of D4Z4 repression, the kill switch model, might be the mechanism of silencing initiation at the D4Z4. Furthermore, this region also overlaps with the region previously identified to show hypomethylation in FSHD cells, so a lack of methylation in FSHD when compared to unaffected controls, indicating that this sequence might be a key regulatory sequence for controlling DUX4 expression by seeding silencing. We then sought to determine how D4Z4 segment 5 is being repressed. For time, I won't show the data in this presentation, but we found that both DNA methylation and repressive histone modifications contributed to transcriptional repression through D4Z45. And this was in a transient manner, indicating that D4Z4 segment 5 may be a key site of initiation of repression of the D4Z4. When you take away that methylation or that those repressive histone modifications, the cells will gradually re-silence through that segment. In order to identify the important machinery involved in this repression, out of the many, many known D4Z4 regulators, we chose a panel of factors for a focused siRNA screen in our D4Z4 segment 5 reporter system. I screened several NERD and CAF1 complex factors and some of these other factors shown here. Again, for time, I won't show the data here, but we found that the loss of SETDB1, ATF7IP, SYN3A, 3B, or LRIF1 in these D4Z4 segment 5 cells increased GFP reporter expression, indicating that these are involved in repressive activity through this sequence. We then wanted to see if the D4Z4 segment 5 reporter could be a useful tool for screening compounds that may aid in repressing DUX4 expression in FSHD. We screened several candidate FSHD therapeutics to see if they decrease expression through this D4Z4 segment 5 sequence. We utilized a live cell imaging system to detect GFP fluorescence over 6 days of drug treatment. Promisingly, P38 inhibitors showed a marked reduction in GFP fluorescence in D4Z4 segment 5 and LRF1 cells, and not in control cells, indicating the P38 pathway may be involved in transcriptional regulation through these sequences. Initially, P38 inhibitor treatment suppresses GFP expression in D4Z4 segment 5 in the blue here, and the LRF1 in red. After 72 hours of P38 inhibitor treatment, though, reporter expression starts to recover in D4Z4 segment 5. This mirrors a recent study where P38 inhibition was effective at inhibiting DUX4 in early myogenesis but not late myogenesis. This experiment shows that this platform can reflect previous studies and can be used for long-term drug screening of candidate therapeutics. In conclusion, we wanted to help deconvolute this complex array of epigenetic silencers and hopefully find some mechanisms of silencing initiation and maintenance at the D4Z4. So in this study, a silencing reporter system identified one discrete kill switch sequence in the D4Z4 segment 5 that is sufficient to drive silencing activity. Both DNA methylation and histone modifications contribute to silencing through this sequence, and we identified several factors that initiate this silencing. We also found that this reporter system provides a simple, robust platform for screening FSHD therapeutics that could enhance DUX4 silencing. Leveraging this platform for future studies, we started preliminary work on using this platform for identification of new DUX4 repressors through mass spectrometry and CRISPR screens, and larger scale drug library screens for therapeutics, and we hope that it will be a useful tool for the field. Switching gears, I am now going to briefly talk about my postdoctoral work. The Chamberlain Lab has long been a pioneer in the field of systemic gene therapy for muscular dystrophies, including techniques for gene replacement, gene editing, and RNA interference targeted specifically to muscle cells. My current projects in the lab are focused mainly on the gene editing arm of these adeno-associated virus or AAV-mediated gene therapies. There are several challenges to AAV-mediated gene editing that are currently being addressed. The first is increasing efficiency of AAV trafficking to the correct cell types, which is applicable to all types of AAV gene therapy. There has been great progress in developing tissue-specific capsids such as AAV-MYO, which has a higher affinity for muscle than for liver. Restriction cassettes, or promoters that drive expression of cargo only in restricted cell types, are consistently being improved for higher specificity. Another challenge is editing efficiency, as correct CRISPR editing in cells is usually extremely low, in the ballpark of 1% in in vivo systems. Optimization of specific cell targets and edit sites can increase this efficiency. Additionally, a dangerous challenge to AAV therapies has always been immunogenicity. Introduction of AAV capsid proteins, bacterial gene editing proteins, and full-length protein products that have not been seen or tolerated by the host immune system can lead to a robust and harmful immune response in patients. current work to identify the most immunogenic portions of proteins to potentially de-immunize them, as well as efforts to limit the amount of immunogenic protein expressed, are avenues to address this issue. My projects focus on these two avenues in the context of gene editing in muscular dystrophies. My first project seeks to optimize an inducible gene editing AAV system in muscle cells. Because Cas proteins are extremely immunogenic, they can lead to harmful immune responses and immune rejection of all of the edited cells. Continuous expression of Cas only exacerbates this risk of immune response. We believe that restricting Cas expression to only one brief pulse will allow for editing and decrease this window of opportunity for immune response. After testing several inducible systems, poly-A regulators have shown promise in restricting gene expression to only when tetracycline has been administered. So without tetracycline, a premature poly-A site prevents transcription of the gene. Upon tetracycline treatment here, this poly-A site is masked and the gene can be produced. My goals are to test this system in MDX mice, administering this AAV and tetracycline to fix dystrophin mutations only in muscle cells with a brief tetracycline treatment. I will then measure editing efficiency and immune responses with the hope that this temporal regulation reduces immunogenicity and increases editing efficiency. My second project focuses on studying muscle satellite cells, the main drivers of muscle regeneration. Most muscle cells are postmitotic, meaning they will not divide again. It is the satellite cells that, upon stress or injury, move from a quiescent to an activated state to self-renew or proliferate into myogenic progenitors that divide, differentiate, and fuse to regenerate muscle fibers. In dystrophic muscle, consistent fibrosis and inflammation pushes satellite cells into exhaustion from too much cell division, and depending on the dystrophy, leading to impairments in self-renewal, proliferation, differentiation, and/or fusion, leading to fiber instability and loss. Therefore, gene editing in satellite cells specifically would allow for regeneration and restoration of healthy myofibers and continue to produce unaffected fibers upon subsequent injury. Historically, gene editing in satellite cells has been extremely difficult, however. My goal then is to further characterize different populations of satellite cells to identify better avenues for gene editing. We, in the Bengtson lab arm of the Chamberlain lab, performed a single-cell multiome analysis of satellite cells harvested from young and aged wild-type and MDX mice to hopefully characterize populations more amenable to tailored gene editing strategies. Multiome analysis gathers paired gene expression and chromatin accessibility for each individual cell, allowing for detailed analysis of cell state, cell markers, and differences between populations. My goal is to find new satellite cell-specific promoters, cell surface receptors, or sites susceptible to gene editing or epigenetic reprogramming. We hope this will lead to higher editing efficiency in satellite cells and ultimately better, longer-lasting muscle regeneration outcomes. These tools can then be tailored to other dystrophies, such as FSHD. I would first like to thank the Tapscott, Bengtson, and Chamberlain labs for this work, as well as my funding sources, including the Friends of FSH Research, Chris Carino Foundation, and the Seattle Wellstone Muscular Dystrophy Fellowship. I will now take any questions.
Okay. Let's all congratulate Ellen for having to watch herself give a talk. That's a very brave thing to do. I'm going to give this mic to you.
Okay. Yeah, that was tough. I just first wanted to thank the organizers for inviting me to give a talk. I'm honored to be here.
Thank you for being here. So I have questions for you. Our audience might as well. So I'll give you one and then we'll pass around the mic. So the goal of your first project was to determine which parts of the DUX4 gene are responsible for DUX4 silencing and which pathways are responsible. How could answering these questions help us design new or better gene therapies or genetic medicines for FSHD?
Yeah, so by identifying D4Z4 segment 5 as this kind of hotspot for modifications, we can then use gene therapy strategies to target CRISPR-I, so inhibition to this area, or like Dr. Jones was talking about, DNA methylators to that area, chromatin modifiers, as well as identification of these other silencing factors. If we can boost expression of things that can silence the D4Z4 in this region, that might have some therapeutic benefit.
Thank you.
Hi, first, thank you as a patient for your interest in FSHD. So I did read your paper, and I was interested as to why you screened for berberine in particular. Like, what made you interested in that?
Berberine is a known G quadruplex stabilizer. So G quadruplex sequences form this very complex structure, and the D4Z4 has these G quadruplex sequences found throughout the region, or at a couple places upstream of the DUX4 coding region. And so there was a paper identifying those pseudo G quadruplex sequences, and so I figured I would try berberine to see if that has any therapeutic benefit by stabilizing those sequences.
Another question we had was, can the TET inducible system that you're using in your postdoctoral work, can that be used in patients?
We would like to think so, eventually. Yeah, tetracycline can be administered to humans at safe doses, and yeah, the goal is first to see if we can get it to work in mice, but we hope so.
Excellent. One more question?
I understand what you went through.
It's painful.
I had a question about your dead Cas9, or dead, not dead, but your Cas9 project. You know, I tend to agree with you that you want to limit the amount of Cas9 expression in a muscle fiber, maybe in other cell types. But yet there are patients now, I guess, that have been dosed with Cas9 that are walking around, and you know, seems to be sticking there. What are your thoughts? Like, how should we be thinking about this for the future?
I think it's very important to test patients beforehand to see if they are reactive to Cas9 protein or anything with the capsid proteins. I know that's been kind of the hard part about gene therapy for a long time, is wanting to avoid that severe immune response. And so it's great that it doesn't seem to be, you know, inducing this response in some patients. But we're thinking if we can see the same amount of efficiency, editing efficiency, in a system where it's on once and then it's not on anymore, that could only be beneficial. But yeah, the efficiency is what we're working on now.
Great. Thank you. Let's give her another round of applause.
Thank you.
Okay, so for the last part of our program, we're going to share a quick update from our partners at Springbok Analytics, which is a biotechnology company turning MRI data into high-resolution insights with pixel-level precision. I'm going to give a shout-out to Springbok's Chief Scientific Officer, Dr. Sylvia Blemker, who's here. And she will be available, I'm sure, to chat afterward if you have questions about how MRI is being used, especially in FSHD clinical trials, and to better understand the disease after the event or at tonight's auction.
Muscular dystrophy is an awful disease with no cure, at least not yet. But new 3D AI technology is bringing hope that effective treatments are just around the corner.
Okay, and with that, we will officially wrap up our 2026 research update meeting. If we could please give a round of applause to all of our presenters today. I'd also like to again thank Sanofi and the Muscular Dystrophy Association for sponsoring today's event. Thank our AV support staff from Hardcastle AV. This is the first time that we've been this professional with our AV arrangements, and we really appreciate their support. I'd also like to thank our all volunteer board and SAB members, some of whom are camped out in the back also supporting us. Who worked to organize this program, and also, of course, the community members and donors who make our work possible. This is the end of our Zoom program. In-person attendees, please feel free to stick around and mingle for a few minutes. This room will be open until about 3:15. And then check in for tonight's Diamonds and Denim Gala, and auction will start at 4:30 in the hallway just outside. So thank you, everyone, for joining us.